Okay, well, welcome everybody. Thank you all for staying after the 2 or 3 day meeting of the PCBC. This is a little bit of an experiment here, a hands-on workshop trying to acquaint you all, I guess, in a little bit of detail with a cutting-edge technology, and trying to do that all in a 4-hour session here. We've got a wonderful set of expert instructors lined up here for you. Thank you to Norma and Neff for putting it all together. Most of them are from Steve Quake's lab, and then a couple of them are from my lab. I'm Mark Krasnow from both of the Stanford hub here. Did everybody get the description of the and schedule for this morning? Everybody has that? Or do we need to make copies of that? You should have gotten it all electronically, I believe. There's 18 people that are registered, and if you're on the yeah, exactly, which has the morning schedule. It's actually going to be in 3 different rooms. Actually, everybody has seen this. Okay, so because it's experimental, we need a lot of your feedback on what works and what doesn't. The 3 sessions in 3 different rooms. First, we're going to spend the first hour just giving you some of the background and some of the fundamentals. And then for the second hour, hour and a half, we're going to move you into the hands-on small group session, where we're going to divide into groups of 3 or 4, we think, and have some stations where you will each move through the procedure and actually see and do aspects of the procedure. That's that cooking aspect of it, where the procedure takes a little bit more than an hour to complete, but you will move through the steps and see each of the steps and just not suffer the long waiting times in between each of the steps. And then, what, pardon?
Like great adventure.
Like great adventure. And then after lunch and for the last session, an introduction to the data analysis, which, as you'll hear, is actually, if you haven't done this already, is as with a lot of genomic type approaches, is the most time-consuming and important, actually, and challenging aspect of it, which maybe you'll get a little bit of feel for during that last hour, hour and a half. So.
It'll be in the room that's like this room, but on the 3rd floor.
On the 3rd floor. And then the 10 o'clock, 10:30 session, or 10 o'clock is going to be in the basement, but we'll all go down there as a group. Key, there's food here that I see most of you have helped yourself, but please, it's informal. Take advantage of the food here so that the neurons are firing at peak speed. And then also, because you're going to have access to all of the experts, each of them who are really the local experts in doing it, who have been doing this, have significant experience. This is a time to be asking questions, especially from the 10 to 11 o'clock hour. This is ask them, when you're seeing the technique, ask them about how you're going to adapt that to your own special circumstances, because there are, even in the discussions at the main PCBC, you were hearing about this types of things like cardiomyocytes. How would you go about doing a cardiomyocyte, because they're so big and hard to capture in the C1 device, and what do you do if the C1 devices are not working for your cell type? And you'll be able to talk to people who have experience with other types of approaches as well. So I think it's seeing it, learning the conceptual background, seeing how it all actually works. If you've actually tried it or have a little bit of experience, or have questions about, your particular situation, system, the questions that you're going to, these are the people to ask. And there's plenty of time now from 9 until 1 o'clock. And the lunchtime, I think, will be a good time to do that as well, because, again, food and plenty of opportunities. And I'm going to let Norma introduce you to the faculty of the course here, who have so graciously dedicated their today and actually tomorrow, because there was overflow for today's. There's going to be another similar course tomorrow. So Norma, do you want to introduce this morning's faculty, or should we do it one at a time? Why don't you introduce?
Well, they can introduce themselves.
Introduce themselves. Okay, that's very okay. You're wired for action. Okay, that's great. don't hesitate to stop, interrupt, ask questions, even in these didactic presentations as well.
I think I'm okay on the power. I want to move this over a little bit so I can reach. Okay. Ta-da. Okay. Hi, everyone. My name is Angela Wu, and I'm currently a postdoc in Steve Quake's lab. So I'm going to be rehashing a little bit of what Steve might have already talked about in his didactic lecture, I think on Monday. So how many of you guys were there for his talk? Okay, so there were some that weren't. So I'll just go at the normal pace. So if you've already seen it, just kind of be patient. And then, as Mark said, just take this opportunity to ask more questions in that case. So.
And so before you go, can you just
Sure.
tell them a little bit about your background?
Oh, my background.
And how you got to be
Like
an
more
expert.
extensive. Okay. So I actually did my PhD also in Steve's group, and for my PhD, I worked on chromatin immunoprecipitation and trying to kind of transplant that assay onto a microfluidic device. So my background is bioengineering, and I was working a lot with microfluidic devices before. And then toward the end of my PhD, I started working on single-cell genomics. And the C1 was just being commercialized, so the lab got early access to the technology to kind of help improve and optimize on it. And so I was one of the first people in the lab to be able to use the C1. And some of the data that Steve presented and I'm going to be presenting is basically characterizing sort of the how the C1 device and its single-cell gene expression measurements compare to some of the techniques that some of you might be more used to, such as quantitative PCR and not specifically microarray, but similar types of measurements, and what are the kind of things that we might worry about when we're doing single-cell RNA-seq. So that's sort of where I'm going to start out with, is like, what are some of the questions that people generally have? And since this work, I've been working on single-cell RNA-seq pretty extensively. I've worked on tons of different types of tissue samples, and right now my focus is working on the colon crypt with Dr. Mike Clark's lab in this building. And so we're kind of studying the heterogeneity of colon crypts and as well as colon tumors. So Mark?
That's perfect, Angela.
Okay.
Actually, and one other thing that I wanted to just ask of the audience is, how many of you have actually tried, we want to get a feel for what level you're at right now, how many of you have actually tried single-cell RNA-seq at your home institutions? Is anybody? A couple of you have. How many are at the other extreme where you have heard a little bit about it but know very little about the approach? Okay. All right. So and then are there any experts here who have done extensively single-cell RNA-seq before who might help instruct the others?
On analysis.
On the analysis. Okay, so some people are expert in different aspects of it, which is great.
Yeah.
Thanks.
Okay, so then I'll just jump right in. So basically my work in the beginning was just to, as I said, characterize what the data might look like for single-cell gene expression. And we compared between conventional tube-based assays with the microfluidic, which is the C1, and then also compared directly single-cell RNA-seq versus single-cell qPCR, and of course with single cells compared to bulk as well. So I think everyone's pretty much familiar with why it's important to do single-cell, and I think this figure, which comes from a 2012 Cell publication, is a pretty good illustration of what you can do once you have a sort of a cell lineage map of the gene expression characteristics for a particular organism. So what they did here is for C. elegans, since we know how C. elegans, every single cell, where it originates from in its lineage, they did single-cell gene expression analysis using this panel of 8 genes for every single cell, and then sort of the color represents the sort of intensity of that gene expressed in that particular cell at that stage. And so like, after you've constructed this map, then you can imagine that you can pick a C. elegans worm, pick any cell from it, and then based on the expression of these panel of genes in that cell, you would be able to kind of identify what part of the lineage that cell comes from. So you can see that that's very powerful, and if we had that for human organs, as some of us are already trying to do, it would be incredibly useful. And also, I think a lot of the people that want to do single cell are also doing it because you can do it for rare and sort of valuable cell types where you can't get a huge bulk of cells to do bulk RNA-seq. So a lot of the questions that I get from people about single-cell RNA-seq is, how quantitative is it? How accurate is it? Can we really trust the data that comes from single-cell RNA-seq? How sensitive is it? And a follow-on to that is, how deeply do I need to sequence? Every time I talk about doing single-cell RNA-seq for a particular project, there's always like, well, is it going to be exhaustively expensive, prohibitively expensive? I only have this much money. So the answer to that is actually you don't need to sequence that deep, and I'll show you the data for why we believe that's true. And then also, do you lose any information if you don't do bulk sequencing? And finally, what's the technical variation, sort of what are kind of the noise profile of single-cell RNA-seq? What is kind of the biological noise versus the technical noise, or what else do you see in there? So this is the experimental design that I did. So on the left here, you have all the single-cell experiments. So here's the tube-based stuff. Here's the C1 stuff. So we kind of did a very exhaustive comparison of everything available.
Just
Sure.
on the bulk versus smart, or how do you make sure you're doing so take the kind of kit variables out of the equation. How do you focus on that? Basically, smarter in bulk. Is that
Yeah. So here you can see we did 2 replicates of just like bulk and then using the smarter kit. Yeah. Okay, so the microfluidics workflow, for those of you who have never seen the C1 or done the C1, what basically happens is you start out with your tissue, you digest it or process it somehow into single-cell suspensions, and that single-cell suspension just gets directly loaded onto a microfluidic chip that looks like this. Inside the chip, the cells get captured in this little butterfly-shaped channel, and there's a little chamber right here. You can't really see it that well in this picture, but the cell gets kind of captured in that chamber. And then inside the chamber, you load all the reagents and everything. So that's done completely automatically by the C1. And then inside the chip, what happens is you do the lysis, you do the RT, and you do the amplification. So what you harvest out of the microfluidic chip at the end of the day is your amplified cDNA. And then you prepare the library on the benchtop as you normally would. So for us, since you get up to 96 cells captured, we just do a 96 Nextera library prep on a plate. And then that's ready for sequencing, and you do the sequencing as normal.
Quick technical question. If you had a C1 at one facility and you're maybe wanting to do the sequencing in library prep at another facility, is that something that's reasonably doable or problematic?
Yeah, definitely. So when the stuff comes out of the chip here, it's basically, you multi-channel pipette it into a 96-well plate to harvest the stuff off the chip. And then you can seal it, spin it down, take it wherever you want. Just keep it on ice or whatever it is that your standard practice is.
Are you going to talk about some of the steps in the workflow there? One thing that we looked at in some detail is the differential Nextera step to see how we could optimize to, sort of in light of some difference we saw in.
There you go. Norma says she's going to talk about it later for hers. Yeah. And so here's an image. So the microfluidic chip is see-through. So after you do the cell capture step, you can actually image all your chambers to see how many cells you've caught. Here I've also done a live dead stain to check whether the cells are viable. So in this image, we have a nice live cell right smack in the center of that chamber. There are 3 different chip sizes that you can do. This one is a medium chip, so we were capturing colon cells. So the diameter here suitable is 10 microns to 17 microns. You can have a smaller chip which captures less than 10 microns, so I think 5 to 10 micron sizes. So that's good for a lot of blood cells like B cells, T cells, things like that. And then on the other end, you have the larger chip, which is 17 to 25 microns. So that's good for, you can capture things like macrophages, and I think hex cells might be a large cell size as well. Okay, so let's look at the correlation between single cell qPCR and RNA-seq, which kind of reflects the accuracy. Yeah, sure.
This technique is not applicable for mature red blood cells, right?
Mature red blood cells? Yeah. Have you guys done that?
Only the nucleated
Yeah, because there is
erythrocytes, because after that, the red cells really don't have any RNA.
Well, reticulocytes have an infinite quantity of RNA in it. It's a decay curve. There's
It's all
stuff in there.
hemoglobin, though, mostly.
Well, there's a lot of helpers to making hemoglobin.
Yeah.
Is your question based on you want to capture them, or you're afraid of contamination from them?
no. basically to identify mature cells like red blood cell if the system is not useful for that.
I see. But since you brought up red blood cells, so red blood cells are actually pretty small. And for most of the cases, like when we get primary tissue, there's often contamination with red blood cells. You can do a treatment beforehand using the lysis kit, but sometimes you still get some in there. But the thing is, if you're working with the medium or the large chip, because of the size selection, the red blood cells actually don't get captured. So we don't get very much contamination in those cases from red blood cells.
How far have you guys gotten in terms of just sort of seeing what the population distribution capture rate efficiencies are as a function of small, medium, large chip?
I don't think
you can get some small cells on the medium and.
We haven't done like an exhaustive comparison of like distributions, but we've done enough chips that we've noticed if you have enough of your target cell size in there, you'll get 0 to like maybe 1 or 2 captured that are of a different size, like completely different size range. Noticeably not fitting in the chamber or noticeably much smaller. Obviously, if your target sized cell is kind of very low in abundance, then you're going to capture a lot more of the other ones. Oh, please, come on. But we have noticed that the size selection is actually pretty stringent. So if you have the wrong size, then the capture efficiency will be much, much lowered. So if you have trouble capturing a certain cell type in the chip, and the capture efficiency is extremely low, like less than 20 cells per chip, then you may want to consider going back and measuring your cell size, making sure that you're using the right chip.
Question, is the highly expressed gene that you're showing this correlation with?
Are you asking which genes I'm plotting here? It's a pretty broad range. So what we worked on here were a cell line of colon cancer cells. And the 48 genes that are shown in this graph are basically genes that we curated for that particular cell type, that we knew certain genes should be on and certain genes should be off. So that was the panel of qPCR genes, and then obviously we just picked out those genes from the RNA-seq data for comparison purposes. So there is actually a pretty broad range here. Everything's kind of on a log scale. So the range of expression values is quite broad. And then a bunch of stuff down here that you see are basically the genes that we don't expect to be expressed, so they're very low. But in general, you can see that the correlation is very good. Both the Spearman and Pearson correlation coefficients are quite high. So the accuracy of single-cell RNA-seq is pretty comparable to that of single-cell qPCR. And these 3 different methods are basically different platforms and kits. So the Smarter kit versus this Transplex kit is actually from Sigma. I don't know if they still make it, but it's a single-cell WTA kit. And the difference to note here is that the slope is slightly different. And what that tells you is that for a certain fold increase in gene expression that you're reading in qPCR, you get a certain fold increase in gene expression measured in RNA-seq. And ideally, you want that slope to be 1 because if it's 1, that means that for the same fold increase in qPCR, you see the same fold increase in RNA-seq. And with that, we get the best results in the microfluidic platform. And we think that might be due to reduced biases in the amplification because you shrink down the volume so much. I think, Brian, are you going to talk a little bit about that or no? No? Okay, not at all. All right. You just have to take my word for it. Okay, so also people have asked, what's the limit of detection? So what we've done is in each experiment, we spike in these ERCC RNA spikes, which are synthetic sequences that sort of have a broad sequence composition, different lengths. So they're supposed to be sort of representative of a good number of transcript that you would get in your endogenous kind of cell environment. But these don't have any homology with mammalian genes, and they're derived from bacterial sequences. So if you're doing bacteria work, you might want to be careful of whether you use the ERCC sequences or not. And they come in a series of different concentrations. So we're doing the RNA-seq, and these ERCC sequences get sequenced as well. And we measure their concentration in each chamber. And this is a graph showing what the expected concentration is, because we know what concentration went in, and what the readout for the RNA-seq is. And you can see that the correlation is extremely good. and also that the detection doesn't really fall off until this very low end of concentration. And what we did next is look at where the signal starts to fall off and see what is that limit that we can detect here. And it turns out that the limit of detection is about 1 molecule per reaction chamber. So that's kind of the limit of detection that we have for this system. Now, this is where it starts to fall off, and you say, why does it start to fall off? Well, that's because when you have an estimated concentration of 1 molecule per reaction chamber, by just randomly distributing those molecules across all the different chambers, some of the chambers will get 1 or more than 1, and some chambers won't get any, just kind of probability, right? So the maximum that you can expect to detect is that 60% of the chambers will have 1 molecule or more, and you'll detect it. And our detection rate that we measured here is that we detect 1 molecule at this concentration. We detect it in 40% of the chambers. So that means that we're getting pretty close to that theoretical limit of what's the maximum number of things that you can detect. Okay.
Totally amazing.
Yeah, it's pretty awesome.
At what step do you add in the RNA?
Good question. So what we do is we make a stock of ERCC spikes diluted to some concentration that you're going to be using quite a lot. You add that into your lysis buffer. So you put the lysis buffer onto the chip along with your cells in different little wells. And then once you pop the chip into the chamber, the lysis reagent gets delivered into all the chambers to lyse the cells, and that's how you're introducing the spike-ins.
What about using FPKM? I'm thinking that
Yeah.
that's kind of probably not the optimal way to normalize.
Right. So when I did this work, FPKM was sort of the industry standard, right? Everybody was using FPKM for RNA-seq. So to make things kind of comparable, I used FPKM. But later Ben will talk about FPKM versus counts and why we've almost completely switched to counts right now, because it's a better way to quantify for single cell applications.
Maybe define FPKM for some of the new people here.
What do you say you need?
Sure.
Yeah.
Just moles. We need mole counts.
Number of molecules.
Yeah. Okay, so FPKM represents fragments per kilobase per million. Per kilobase is the length of your transcript, and per million is the number of sequenced reads that you, or the number of mapped reads that you got. So it's normalizing for how deeply you're sequencing and normalizing for the length of your gene or transcript. And I guess Ben will talk about why it no longer really makes sense to use that metric for single cell applications.
What if you have vastly different cell sizes in the prep that you want to look at? Do you just use 2 chips of different sizes?
That's a really good question. So I think so far that's what we've been doing, is just separating the 2 different, or doing it twice on 2 different chips. Yeah. Some people also do like a round of fax sorting first, and they sort by size on the fax and then run 2 different chips on, so that you get a higher kind of rate of capture.
It's a very different situation though when you do fax purification, because then you kind of lose a lot of points of reference in the data set.
Well, you can sort in the fax just by size without using too many surface markers, right?
I know, but everybody's tempted to like I just want to understand my population, and they don't realize that you can understand your population a lot better if you have some stuff to compare it to that's not like yours.
You had another question back there?
Yeah, just about the depth of sequencing. Do you see the same number of reads across the board, like 50 million reads for everything on one chip? Or do you see a lot of variability?
Per cell variation in read depths.
I think I'll show some data that will sort of answer that question. And if it doesn't answer it, you can ask me again.
Is there a way of checking the quality of RNA and your input?
Well, you're inputting like whole cells onto the chip. So the live dead stain is one way to see if your cells are viable. But after you get the cDNA, we typically run like a fragment analyzer, which shows sort of the cDNA quality that you get out, whether there's degradation. And I think someone will talk about that later today in more detail, like sort of the whole QC. Yeah.
Your cDNA
right.
in the reads.
Okay, so
But this is a big deal, this slide that you're showing here, to
Yeah.
just explain what's going on.
So here we wanted to make sure that we didn't lose any information by just looking at single cells and not doing the bulk sequencing. So what we did here is on this axis, the horizontal axis, what we plotted is we took all our single cells, the reads from all of them, then bioinformatically merged them all into one pool, and then redid the whole alignment, trimming, all the quality, pre-processing of your sequence reads, and then mapped them the same way that we normally would for, for our single cell reads. So this is sort of what we call an ensemble, or a mock bulk, if you will. And on the y-axis here, we're plotting the real bulk RNA experiment. And these are from the same cell type. And you can see that, and each dot represents a gene, and you get gene expression from the mock bulk versus the real bulk. You can see that the correlation is really high. I mean, it's like 0.9. So that means that you can totally recapture whatever you see in the bulk by doing single cell sequencing and then mashing everything together. And what this tells you is that the bulk really kind of is really an average of your cell population, and this kind of really illustrates that.
If you're missing, and I've looked at data where we've done something similar, and we're actually missing a lot of the high-end stuff in the bulk that's from the single cell when you merge it.
High-end, as in from this corner?
Yeah, and I mean, it's not like a large proportion necessarily, but it's it's a decent number of events. If that's happening, is that indicative of something problematic that you would know of?
So you're saying you see it in the bulk but not in the ensemble, or the other way around?
You see it in the bulk but not the ensemble.
Well, actually, Nathan, we don't have a comparable situation because most of what we've done is Ovation V2 versus Smarter. And so there we're picking out stuff where it's just Smarter's not able to make cDNA out of it.
I see. It could be the poly A or whatever else. Yeah.
I think one of the things, keep the questions down, because a lot of people here have no background in also
Sorry.
a lot of the terms you're going to have to define, or save some of those specialized things for the end.
It's the kit variables. So the kits are really able to pick up different RNAs and got to be sensitive to them.
Okay. So are there any questions on the, like, if you're completely lost, please let me know. Okay, so this is, in this graph, I'm trying to show what the sort of variation is between samples. So here we have the gene expression level of the genes, and then on the y-axis is what I measured as the median absolute deviation. So we use the median instead of the mean, just because the median is a little bit more robust. And what you should think of it as the coefficient of variation, sort of plotted on here. So the noise level you would think should decrease as the gene expression level goes higher, because if it's a really highly expressed gene, you should expect to see it robustly expressed over many replicates, right? So here in the bulk samples, you can see that's exactly what you expect. The noise profile kind of dips down like this. And with the single cell case, it looks very similar in sort of the profile and the way that the noise decreases as the gene expression level goes up. But what you're missing is this bottom corner where there are low expression genes that are also being robustly expressed in replicates. And that makes sense because in single cell cases, each single cell is different, and you're going to have bursting and things like that. And genes that are expressed at a very low level may be captured in one cell but not captured in another cell, which causes that higher variation for low expression genes. And here again, you see that mock bulk or ensemble experiment. It looks identical to the true bulk experiments. And this is the tube-based format compared to the microfluidic format here, where you see that we actually lose some of the dots out here, or we get more dots out here where highly expressed genes are more variable, or seem to be more variable, right? So actually the C1 gives you sort of less variability for the genes that are more robustly expressed.
Is this excluding just like dropout events and genes that were just zero?
I think this includes them. They would be, if they were zero in some cases and not zero in other cases, they'd be, up here. Yeah. I think this is including everything. So all zero stuff would be like down here. All right, so this should be addressing the question about sequencing depth. What we did here is for each bulk experiment and also for single cell experiments where we sequenced more, we bioinformatically downsampled to fewer and fewer numbers of reads to see how many genes we would detect at each sort of depth of sequencing, if you will. So here you can see that it actually saturates very quickly. The single cell experiments are down here, the bulk is up here. And so actually, if you zoom in on this lower portion, you can see here, you saturate very quickly. With the single cell experiments, by about 1 million reads per cell, you're getting all the genes that you're ever going to get. Why do you get so many more genes when you do the bulk? That's because, again, you're mashing all the heterogeneous different cells together. And so, of course, you're going to see some genes that are not represented in all cells, but in some cells. So you see that in the bulk. But in the single cell, you might just miss that one. So the key here, though, is that it saturates at 1 million reads. And you probably don't need to go that much higher, like to 5 million or 10 million reads, to get everything that you need to get.
What about read length and paired end and junction counting?
I have not. Did we do anything on that? I don't think we looked at junction counting for. These are all, or some of these are single-ended reads, but I don't remember which one. But most of these are paired end, and I think right now we prefer to do paired end just because you get better alignment, you get better mapping, and for.
We'll go into that more this afternoon.
Yeah. So a lot of the basic sequencing metrics that you're thinking of, like what's the coverage like and things like that, I think we'll talk more about later.
But just in the conclusion to this kind of study, though, you want to know what's your likelihood of detecting splice forms as a function of read depth too.
Here we didn't look at splice forms at all. We're just interested in gene level characterization. Okay. So this is sort of just a conclusion to everything that I said. Single-cell RNA-seq we found is quite accurate compared to the gold standard qPCR. And the sensitivity is much higher on the microfluidic platform because also you have less bias, and we see that the limit of detection is very low. And then you don't lose anything when you don't sequence in bulk. As long as you have enough single cells, you can mash them together to recapitulate what the bulk looks like. And to know how many cells you need to do that, there's been recently another paper published by, I think, Fluidime and Arnold Kriegstein's group, where they illustrate some of that. So you can look at that paper. I think it's in Norma's list of references. And then I showed you that the sort of technical variation, and it's very reasonable for what we would expect for single-cell RNA-seq. So the conclusion is that you guys can all safely use single-cell RNA-seq without worrying too much that you're missing out on a lot of information from bulk studies. And I think later people are going to talk about what sort of biological applications. And yesterday, I'm sure at the conference people have also talked about that. So I'll be around if people have additional questions, and then I'll just put up the acknowledgement slide for all the people that contributed to this work.
Is this tool useful for identifying like novel genes?
Novel genes?
Yeah.
So I think in our lung lineage paper, there were some, were there novel isoforms or
There were novel.
Yeah, novel isoforms that you can find using single-cell RNA-seq. And some of those isoforms are very low in abundance, and so if you looked at them in bulk, you might not pick them up, and you might not notice the sort of cell-specific isoform, splicing type of characteristics. Does that answer your question? And I don't think we've tried de novo stuff, like de novo assembly of like unmappable reads or anything like that, but yeah.
What about increasing the likelihood of getting to the true 5' end? Have you looked at factors that can improve the sort of detection of the 5' most exons?
I think that's more a biochemistry problem, less like a platform problem. Yeah, and so we're going to leave that to Clone Tech or NewGen or whoever.
And then what about in your bulk versus single cell merge comparison? Did you know that the situation that you were comparing there, you didn't have cell type sizes that were going to be missing from the capture?
Say again, sorry.
So if you do that, if you try and do the bulk versus merge comparison, but you have a cell size subpopulation in the bulk that is going to not be well captured on the chip, then that would show up as a underrepresented fraction of genes.
This particular data was done with a cell line, so there wasn't cell size differences. But I mean, I would think that may be the case. We have data from different cell sizes. We could probably look at it, but it's not a really high priority at the moment. Yeah. Is anyone completely lost and need to clarify things? I just want to make sure that the subsequent talks all make sense as well. Okay, great.
So for the beginners, everything's okay? Speak up now. Okay.
Yeah.
This is a great presentation. Thank you.
Yeah, sure. Oh, and I should give you the.
There's more coffee and breakfast.
After the question. No, I'm good. I'm good. I don't know. I'm good. It's designed for.
It's not like it's not.
Okay. All right. Wait till everybody's done. Huh? Sorry. Oh. Whoops. Okay. Oh yeah, sure. Okay. So good morning, everyone. My name's Brian Yu. I'm a PhD student in Steve's lab, and I've been using the C1 extensively to. So before, when I first started, I was working with microfluidics, doing microfluidic cell culture, working with a high throughput automation around this microfluidic cell culture platform and looking at phenotype differences. Then the C1 came along, and then now I'm doing using the C1, kind of adapting it for different purposes other than RNA-seq and other than mammalian cells as well. So I've been using it to do kind of environmental sample metagenome kind of thing, looking at trying to assemble de novo assembly of DNA sequences from bacteria, and also yeast RNA-seq, fungal RNA-seq from environmental samples. So today Norma asked me to talk about a little about microfluidics and also the C1 script generator. So how this fits in with the grand scope of things in the workshop is kind of like, let's say we all know that single-cell RNA-seq is great, and we all kind of want to learn how to use a C1 platform to do single-cell RNA-seq. So hopefully my slides will show you what the C1 platform can do, what it comes in as out of a box, and what you can change on the C1 platform to adapt to your particular needs. So let's first take a step back and look at what technologies is the C1 platform built on, right? So microfluidics, multi-layer PDMS microfluidics is kind of like the technology that enabled the C1. The material that the microfluidic chip is made out of is a polymer called polydimethylsiloxane. It has certain properties, physical properties, that made it very suitable for doing cell work and also kind of for microfluidics. One is that it's viscoelastic, and that's going to be important for these type of valves because it's flexible, basically. It's transparent, which makes it possible for you to image it when you have cell captured and show like great images like Angela just showed. It's also gas permeable, which is basically good for a hydraulic system. So think of the chip as a hydraulic system instead of a pneumatic system. There's no air whatsoever inside the chip. And then finally, it's biocompatible, so then your cells don't die even if you do some kind of stimulation on the chip, just like there was a paper from Broad Institute recently where they did stimulation of the cells and then RNA-seq, right? So there is one element on the chip that basically makes it very powerful, makes it capable of doing 96 replicates of the experiment at the same time, and that's basically this push-up valve. So how you look at this would be, this is a flow layer where your cells would go through. This is a control layer where it could regulate the flow of the reagents on the first layer. So these are channels that goes into the page, for example. And then finally, you have the glass, or in the C1 case, it's a plastic, transparent plastic plate, right? So here's a top view of how a valve is actuated. So basically, think of it as you have a garden hose that you're watering your lawn, and then you step on it to stop the water flow, right? So that's exactly what's happening. So here you have a control channel that's blocking out the underlying flow channel or overlay and so on. So on the so this is basically.
I've never seen this explained so clearly.
Okay. All right. Thank you. So this valve basically enables all the functionalities of the C1 chip, and it's used mainly in three ways. So the first way is to isolate reaction chambers. So when you have a cell mixture flown onto the C1 chip, it isolates the cells into single cells, and that's the geometry of the capture site. But once you do that, you want every single cell to be isolated during the entire RNA-seq protocol, and that's done with these valves. right? Another use of the valve is to regulate the flow of reagents. So in here, how it works on the C1 is that you have 8 reagent input. This is where you put in your smart clone tech kits, maybe the first step like synthesis, first strand synthesis, the RT, the PCR, and so on. And actually, all the reagents come in through the same channels. So how does it regulate that? So here's an example showing that basically you have valves blocking each of the input. And do you want to click? And basically, one valve would open up, and the reagents from that particular inlet would flow into the chip and get introduced to all the reaction chambers. And the final use of the valve is actually to move reagents around on the chip. So if you put 3 valves together and kind of actuate it in a certain sequence, it could actually, basically due to the asymmetry in the actuation, it would push fluid from one side to another. So that's how actually the reagents are introduced on the chip. So now that we know a little bit about microfluidics and how the chip actually works, so here is what the C1 comes in as their kind of out-of-the-box default software, right? Using Smarter Kit, you can only do certain number of PCR cycles at certain temperatures. Nothing, you can't really change anything. It's really easy to use. There's like run, that's it. So the first thing you do is what they call priming the chip. And that's basically, if you think of the previous slide, I had a picture of the push-up valve and there's this control channel. So that's basically filling the harvest reagent into the control channel or buffer into the control channel. And the control channel are basically outlined in red here. So this is the overview of the center portion of the C1 chip. And the second thing you want to do basically is loading, and that's putting your cells in. And what's actually happening in a cell is that cell goes in one end, goes around. So this is from, this is capture site 48. It goes all the way around all the capture sites and comes out the other end, right? So here is if you run, so basically it comes in capture site 48, goes to 1, goes to 49, and goes to 96. So if you notice, if you run a lot of chips, you would notice that if a chip gets clocked, it clocks at capture site 48. And this is the reason why, because it comes in first. And this side of the chip always gets more captures than this other side. And the reason is because it goes in here first and then comes out here, okay? So that's basically what's happening. And then since this step on, all the reaction chambers are separated and the cells don't see each other ever again. Literally, oh no, that's right. And then, so the third step of this default protocol is DNA synthesis, and that's basically according to the Smarter Kit. And all the reagents come in through the middle channel and gets pushed out, right? So each of the reactions happen in a horizontal fashion. And at the end, you collect your samples out here and out here. So that's the default. And we all know that default is never good enough, and we're doing experiments. And the one key thing about experiments is we want to change things, right? We want to adapt new. We want to change the PCR cycles, for example, change temperature. We want to adapt new reagents, new kits, new cell types, and so on. So in fact, do you want to click it again? So I like to think of a C1 chip as just a tube, basically 96 of these tubes, where for each tube you can add reagents 5 times. Okay, right, so exactly that. And Fluidigm luckily makes a software interface for you to do that, to change the C1 protocol, change what it does. And it has 3 kind of interfaces, and I'll just go through the 3 interfaces. It's actually very easy to use. First thing is called this protocol summary. So here is where you set up what you're doing. And they have some default ones where they have the STA, the mRNA, and the DNA-seq. Let's say I dragged one of the protocol on, and you would see that, so I added some extra parts. So you see that the cell load and stain includes the capture, the stain, and the wash. So these are flow-through steps. And what flows through steps is basically the reagents comes in through here or any of these inputs, and it goes from capture site 48, goes around the chip and comes out. So that's what a flow-through step is defined as. And you can add as many of these flow-through steps as possible in this first block of code, right? So you could do staining, stimulation, whatever. The cells will stay. And then the second block of code.
You have to explain that stimulation step.
Well, I don't know, but I mean it's a paper where they stimulated with some kind of
LPS.
Yeah.
So they just flow through LPS through all the channels, and the cells would just.
So for example, the cells are sitting in the capture sites, and then you can. So wash buffer is one thing. That's the default wash step, where you put wash buffer in this well. So let's say the cells come in media, right? or something that might not be compatible with the first strand synthesis of Smarter kit. What the flow through step does is it does a buffer exchange into whatever you want. And typically the wash buffer from Fluidic, right? And then you can introduce the reagents and that will work from there on, right? So think of flow through step as a buffer exchange, right?
But, let's say we wanted to, exactly what do you need to do to do a complete wash and then stimulate, and then could you do 2 sequential different stimulations?
Well, so that you can figure out, but so the script allows you to
Just give
put.
us an idea of what has to go into the script in order to accomplish that kind of thing.
So, okay, so
I think that
that might be a good question later on. So I'll be downstairs this afternoon to run the scope, so you can ask me there. Okay, so the second block of code is basically called sample preparation. That's the individual reactions, right? And that includes a series of these mixing steps. And the mixing steps is basically kind of a selection of one input which goes into the central channel and gets pushed into all the channels. So that's what the definition of a mixing step. And you can put any kind of mixes here, okay? And then after a series of mixing steps, then at the end you do this harvest, which is, it pushes more fluid in so that you have enough volume to collect your cDNA, right? So basically at the end, you're in the first panel where you would see your protocol and basically kind of version control your names and so on. So that's the first interface. So the second interface is reagent assignment. So now you told the script kind of what you're trying to do. You want to do these 5 things or 6 things and so on. Here's where you set what the thermal cycling temperature is, what all the specific parameters. So the first thing you set is kind of the chamber. Where do you want the reaction to happen, right? For example, here, this is a standard RNA-seq, right? You have lysis, RT, PCR. And the PCR is done in these 2 large chambers. And then so you set that. And then the next thing you would set is, okay, so where is the reagent coming from, right? So in this case, reagents are actually coming from here, but so these are all the different places you can assign the reagent inputs. And then the third thing is you would
Just want to briefly say
Yeah.
what those chambers are on the
In the standard RNA-seq.
in the standard RNA-seq what these
What
chambers.
happens in each of those chambers?
Oh,
where
okay.
the cell
So
comes from and
it's
then-
color coded, right? So here is where the cell comes in and gets captured, and then this is where the lysis happens, and the whole thing is where the reverse transcription happens. So it pushes the reagent in, and then it uses the entire volume, and then pushes more PCR master mix in to do the PCR in all 5 volumes together.
Are these different physical chambers? Is the cell moving around, or is this?
So the cell
just
is gone.
kind of
So the
virtual-
cell is done
Okay,
after the
cell
lysis.
detritus. Or are these just kind of a virtual chamber where you're putting different amounts of reagent in? I'm
These
just,
are physical.
These are
These are
physical
physical
chambers.
chambers,
So if you
Because of your valves.
so look at here. So this is chamber 1, 2, 3, and then there's 2 more chambers over here. So this is actually a chip.
Okay.
Cool.
All right.
And so it's like you're changing from one tube to the next by moving the
Well,
valve.
you're actually adding. So that's why I
The
said
tube's getting bigger.
the tube's getting bigger, right? So that's why I made the analogy with a tube, and you're adding a reagent 5 times. Okay. So yeah. So after a reagent assignment, you would set these thermal cycling protocols, right? For lysis, this could just be room temperature or 4 degrees or something. And then for PCR, this is kind of the same thing as the biomark thermocycler protocol interface, if any of you have used that. And then finally here are kind of, I think here, yeah, loading and mixing temperature. So these are the temperatures at which the reagent gets introduced into the chip and gets mixed in the chamber. And that's actually a non-negligible amount of time, which you cannot change. So if you go to the next slide, so this is kind of an overview of your protocol, and that's a summary, right? So you want to, so at here is basically you check that you did everything correctly, right? For example, each of the box here, each step here would represent a block, and this what happens in the block. So if you notice that there is a lot of these reagent loading steps, and that happens for each block. At the beginning of each block. It's not something you can edit or change. And at the beginning, it takes quite little time, but later on, it takes as much as like half an hour, right? So it takes a significant amount of time. And it's important to make sure these things are here. Right? And then the next thing is harvest. This is also something you cannot change. But what you could do is you could kind of elongate this pre-harvest time so that the reaction can run overnight and you can adjust when you want to harvest. Like for example, the reaction finishes at 3 a.m. You don't have to harvest at 3 a.m. You can wait until the next morning to harvest. Right? And then the third thing is that it also tells you for the size of the chip you select, what is the average or estimated amount of time it's going to run the whole script. Right?
Excuse me?
Yeah.
Can you do like gradient, to optimize, the application conditions?
Can you say that a little more specific? So
when we do, for instance, PCR,
yeah.
we need to do gradient to optimize, the optimal, temperature.
So you can do
that?
any, so like a touchdown? Yeah, you can do that. You just have to set the PCR thermocycling protocols here. In fact, this is a touchdown, isn't it?
There's one script for every well.
Right. So every reaction sequence is exactly the same. You cannot
treat
try.
one part of the chip differently.
No. It's very small, and then basically it's sitting on a thermal Peltier heater. So if you can't really heat up one part and have the other part not heating, right? So there are current limitations with the chip. Just one, due to the geometry, two, due to the fact that Fluidigm doesn't want to break their machines. So all flow through steps.
Run the time of chips, just one?
Each machine, yes. So all flow through steps must be performed before any mixing steps. So this is the two blocks that I'm showing, right? The first block includes the flow through steps. And once the flow through steps are done, the cells are isolated. And from then on, you cannot flow through again, right? So these have to all be done before any mixing steps. And the second one is that during flow through step, all capture sites are connected. So if you're not okay with that, then maybe it's not a good idea to do flow through steps. So this is when the cells are still seeing each other, kind of. Okay, and then talking to each other. All mixing steps must be in one section of the script. So basically, let's say you want to do certain reaction steps, right? And then you want to take the chip out, and then you want to put it back in again with some new mixes. Currently, the script does not let you do that. I don't know why. I don't think it's kind of like I don't think it's a technical issue. It's just the script, the software doesn't allow you to do that at this time. So those are the limitations. Other than that, I think anything is free for you to try.
On adjacent cells, are there any measurements of like parts per million of mixing that occurs in?
That's probably very negligible because they're actually really far away in terms of distance. If you look at the width of the channel and the distance between the channel, between the two cells, actually pretty far away. What I mean by they're still talking to each other, it's if you're flowing things through, you can still wash things out without, that's not attached, right? So for example, for me, if I want to load bacteria is too small to be captured, so I don't want to wash, because then I wash everything out. And that's my last slide, actually. So if you have any questions. Okay.
The whole thing was just amazingly clear.
Is there any way to like go
Neff, and I'm the director of our sequencing operations here in the Stem Cell Institute. And so my background in sequencing is that this is my 6th year doing Illumina sequencing. I started off doing a year with Rick Myers and Barbara Wald in the ENCODE project doing ChIP-seq and sequencing on the original GA. And so then I joined the Quake Lab and started working for the Stem Cell Institute, where we branched out into doing a lot of other things. So today what I want to do is just tell you some of the things that we're doing in our facility and to go over some of the new improvements in DNA sequencing technology. So our lab downstairs is we're not a production sequencing facility, we're a protocol development. And so what we've done is to really focus on small numbers of cells and small input library construction, because that's the primary interest of the Stem Cell Institute. So here in the Stem Cell Institute, there are basically two focuses. on small numbers of cells or single cells, and that is for stem cells as therapy and also in cancer stem cells. And so we have this little mantra that one cell will cure you, one cell will kill you. And so our goal is to try to help develop sensitive assays using sequencing to facilitate these projects. So you've heard a lot about the C1 and how it works and the basic parts, and so what I'm going to do is just talk about basically the cDNA to sequencing part of the protocol. So the Smarter kit that we're using is one that takes advantage of a special feature of reverse transcriptases. That is, so after you prime with an oligo that has a PCR tag and an oligo DT tag and make a full-length cDNA, once the RT encounters the cap, it turns around and it makes a small terminal transferase-like activity where it adds on a small homopolymer on the end. And then you get what's called strand switching. You can come in with another PCR tag and another homopolymer that matches this one and then copy it to make second strand synthesis. And so what you end up with is essentially full-length cDNAs with 2 PCR tags on each end. And then there's a PCR amplification to take the single cell material where there's on the order of, say, 10 picograms of RNA in a cell, and find those small number of transcripts and amplify them up so that you can make them into a library. So when you see the C1 chip, the harvest wells are covered up with these pieces of white plastic, and so that's designed to keep contamination out of your harvest wells while you're doing your reagent adding and your cell loading. And what we do is to pipette from these wells into a 96 well plate, which we call the harvest plate. And then this becomes sort of the permanent registry of the experimental material. And then from the harvest plate, we make a dilution plate where you basically do an all-in analysis of what came off of the C1 on the fragment analyzer. And then from that data, you calculate a dilution plate, which is used to make your library construction. So we found the fragment analyzer, the AATI fragment analyzer, to be essentially indispensable in this because you can't capture 80 cells and only look at 10 samples on the bioanalyzer and know what's going on. So the fragment analyzer is a very simple TBE, ethidium bromide, capillary gel electrophoresis system, almost like an old Sanger machine, which displays for you your 96 samples. And so this is basically a trace of one of our cDNAs where you can see there's a lower marker and an upper marker, just like a bioanalyzer. And so this is, of course, a good cDNA prep coming out of a well. And so what you can see are some of the ERCC spike ends that show up during the prep. So one of the things about the spike ends is that it's helpful to know how much RNA your cell has. So we started off with the HCT116 cells that Angela talked about, which actually make a fair amount of RNA, being a tumor cell line. And then we switched to doing B cells, and so it turns out primary B cells and primary T cells have very little RNA, so that some of those early experiments, the reads were 90% spike ends. So every cell has a different amount of RNA, and every cell type you work on, you have to think about calibrating your spike ends, and it's good to err on the side of low at the beginning because otherwise you'll get too many spike end reads. So once you make the cDNAs and make the dilution plate and do the library plate, we find that the dilutions that Fluidigm recommend in their protocol for making the Nextera libraries work really well. So that at the end of the day, when you have your finished library product, you basically just take 2 microliters out of each well and pool them and clean it up and submit it for sequencing. So we find that these Nextera XT libraries work very reproducibly for this. So if you have a good cDNA, you're going to get a good library. And we've been using the new 384 Illumina barcode series to make libraries so they can be pooled in large or small amounts for sequencing, depending on how many cells you get out of your experiment and whether you want to sequence all of your cells from multiple chips together at the same time.
Are
Are there any issues in switching from the 96 barcode to the 384?
No,
Totally.
not that we've found so far.
Don't
Yeah, don't mix the two, right? Because they're different. So they've in the 384 series, they've dropped out some of them. Like 501 is no longer there, for example. They don't. It didn't work as well with the rest of them.
Can this be done?
But-
Is stranded and unstranded?
You would have to go to the protocol generator and use a different kit to do that. Currently, we haven't done too much in trying to, haven't had any success really in trying to get some other protocols to work in it, but it's certainly something you can do to change the protocol and use any other kind of cDNA synthesis that you really want to do. It's pretty flexible. And so,
Have you worked much on the Nugent stuff? Stranded?
no, we haven't gotten the Nugent stuff to work very well yet. The tube control works, but the other one doesn't. Anyway, so this is our facility, and so these are some of the sequencing machines that we currently have or have had or in the past. And so the C1 and the fragment analyzer, we also have a Nugent Mondrian that we've made like 1,500 libraries for our cell-free DNA synthesis using it, because the cell-free plasma DNA comes pre-fragmented. So basically, it's once you get it out of the plasma, it's ready to go in the Mondrian system, and this has worked really well for us. And so these are the 3 sequencing platforms we have right now. We're sort of stopped using the HiSeq 2000 and focused primarily on the 500. And there are 2 key features. One is that the cost of using the NextSeq 500 per megabase or gigabase is basically the same as running the HiSeq. And also it runs very fast and gives good yields. So primarily the MiSeq we use for QCing libraries and also for doing metagenomics. I think I stuttered there anyway. And the NextSeq we use primarily for doing the C1 RNA-seq at 2x75. So there are 2 kinds of flow cells, the mid-run and the high-run flow cells, depending on how many libraries you have to sequence. And so the mid-run 2x75 is the most cost-effective. The kits are only like $900. And you get basically a HiSeq lane's worth of information out of it. So those of you who don't know about the NextSeq, it's actually Illumina's next generation machine, which has a new chemistry. It only has 2 dyes. Instead of it being like the regular earlier Illumina chemistry where all 4 bases each has a different dye and they're added all at the same time, here the G is dark and A is a mixture of the C and the T fluors. So basically you end up with like a fluorescent FACS plot for looking at your data. The NextSeq has a little bit to go as far as like its error rate. So another reason we do a lot of 2x75 runs is because the error rate of it is down here in a happier range, more like the HiSeq, whereas when you get up to like 150, the error rate on the NextSeq starts to go up to like 3 or 4% at the end of the 1x50 runs. So one of the other things you can do that what Brian alluded to is DNA-seq, is you can do whole genome amplification on single cells on the C1, and so it uses an MDA like protocol to do the amplification. And one of the other studies we've done in our lab is to capture single cells and use the Fluidigm access array system to make a lot of amplicons and sequence those for SNPs in a project looking at ALL cells. And so you can find new information, whereas this person clearly has 2 dominant clones in their population, and something we wouldn't have known if we hadn't done single cells.
What about those double positive cells, the 2 in the middle?
Oh yeah, there are a couple of them. If Chuck was here, he would tell you about them, but this is for a taste of this technology. So, so downstairs we have some people who are going to help you look and understand more, answer questions about the different technologies. So Brian is really one of the unsung heroes of this project. He spent a lot of time writing scripts and setting up the microscope. He even built the hard drive for the computer in order to make this work. And so you'll see from what the microscope does that it's really one of the important parts of this process is to being able to capture and image the cells that you're going to lyse and then do sequence. And so we'll also have someone down at the fragment analyzer to give you an idea of how this works if you're curious about this. And Doug is going to talk about tissue dispersion methods. And then Joe and Spiros will be downstairs to answer questions about the C1. And one of the nice things is that Fluidigm lets you run used chips on the machine. So you can load chips and see how it's kind of a black box in its current setup, but please feel free to ask questions of the people who are down there and to look at the equipment, and I can show you some of the sequencing technologies for those of you who've never seen a helicose machine, for example. We have one.
So we are going to get a presentation on tissue dispersion methods?
Yeah, downstairs, yeah. We'll set Doug up in a little conference room downstairs and he can do his presentation if you're interested. Okay, thank you all for coming.
And then we're going to meet upstairs at what time normally?
11:30, 11:45. That's when the pizza's delivered.
I know for
yeah, we do Oh yeah, those are giant. We do like both extremes, right? The stem cells, which are like 5 to 10 microns, and the heads, which are giant cells.
Okay. So yeah, so after it finishes imaging, I think this is like a new feature that is most useful for at least people who are using this software. So basically, after the chip, you allocate things out in different order. So not capture site wise, not necessarily in transit. So basically, what this generates is it generates a big mosaic with this middle section showing every single capture site. And so basically the mosaic looks like this. So if you have a cell in there, it will show. And this is in the format of the 96-bit, right, not in the capture well. So you can just easily kind of pick out, okay, which cell is good and I want and which cell is not. So let's just take a look at other people's.
So just also separate target. So if you meant you're already kind of getting out that stuff already intact from that triggering array, which is a very fancy word for just pipetting up and down, or they're using glass pipe, Castro pipe.
So that's so for example, right, she got all these cells, right, and then the fluorescence overlaid on top, so the green means it's alive, and then the orange means it's dead. So some of these are kind of dead. And then here, you know that you see that, okay, you have some debris, so maybe you don't want to use the data, or you don't want to sequence it over cell. And so on, like here you have multiple cells and stuff like that, so you don't want to use it, data from that cell. So that's why it's very important to have these images captured before.
So this is okay. So now I'm under my pure.
Say again? The green would be kind of like a live state. We have a live dead state. Green is live, the orange is dead. Right. It's in vitro dead state. So another thing is, so you can we've changed it so that you can use user annotation, which is when you're taking the images.
You'll see when you spin down.
Just to summarize. So when you're taking the images, you can user manually input if there's a cell, there's no cell. Later on, you can also use that annotation or change that annotation. So for example, use see there's no cell, so you type no. And then if there
So
is a
what
cell,
about
you
removal
type
of dead
yes.
cells in small
Right?
volumes?
If there's a cell, you type yes. And if there's two cells, you type like maybe or something, so you have yes, no,
but
maybe.
your
And then this is for
choice
each
of
channel. So later on, let's say you
using
pick
magnetic
to take the image using fluorescence, you would basically
allows you to simultaneously get that.
basically say, okay, is there fluorescence? There's no fluorescence. For every channel of the image, you can do that. It takes longer. It makes the imaging process longer. Some people choose not to do that, but
Okay, so yeah, so basically, so we have the pipeline, we have the cell, it got dissociated, we put it on the chip, right? So this station is basically capturing the information of the cells after you load it onto the chip. So originally, when we got the C1, Angela and Barbara was basically imaging the chips upstairs with like Mark and Phantom to kind of like saving the images manually. So it's kind of, we wanted, and meanwhile the cells are sitting there dying. So we wanted a way of automatically imaging the cells from the entire chip. So that's when I came and tried to like write a user interface to do that. So basically the goal is so that later on when we have the sequencing data, we can kind of correlate back into the morphology, well not morphology, basically if the cell is live, if the cell's dead, what kind of, if there is a cell or if there's like multiple cells and so on, right? Maybe there is no cell and that's why we didn't get cDNA, or because the cell is dead, or it's just not producing that much cDNA, right? So the idea of this is basically trying to record as much as data as possible about the experiment and the cells without spending that much time. And the first thing we usually do is here we note down like what tissue is it from, what species is it from, what facts sorter did we use to sort the cells. Maybe like here you can write down things like patient age, patient like organ, like disease, and so on. And then here's like you have your SUNet ID, and then I try to highlight this as a new addition. And basically you just kind of like do this, and then it enters your barcode in here, right? So once you have the chip on the microscope, you first have to initialize it. That's finding the focus of all the XYZ for each of the chamber. So how this works is we are basically, this script basically goes to 3 corners and asks the user to focus on the 3 corners. So that's kind of like chamber 1, 48, 49, right? And then once you focus on the 3 corners, it automatically focuses on 3 other corners, which is the other corner and the 2 middle, right? And it fits 2 parabola. So now the Fluidigm actually makes these chips slightly curved, so we can adjust for that. So I just focus on the 3 corners, and now it's going to go to every single position. Well, not every single position. Go to the other 3 positions and autofocus. And then after that, what people do is they can choose to image in phase and different fluorescent channels, right? So if we're just doing cells normally, we tend to use a live dead stain from Invitrogen. So it would be blue when the cells are alive, and then it would be orange or YFP when the cells are dead. And then at the end of the day, we can have all these images saved automatically and then overlaid on top of each other so that we can tell which cell we want to sequence and which cell we don't. So this is almost done. And once it's done, so one way to do it is just select your imaging method, like phase, fluorescence, the 2 fluorophores, and also different gain and exposure states, right? And after you do that, let's say we just do phase, and then you click run. It basically just goes through all the positions and takes an image. So one thing people are really they really want to know during this step of the experiment is how many cells did I catch? Right? Sometimes if I catch too little cells, I want to reload. I want to load again to catch more cells. So this is a good place to do it. So as the image is going, you count how many cells. So one thing I noticed at the earlier versions of this software, people was taking a piece of paper and noting down like cell, 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 no cell, and then adding all the numbers up. So I thought, okay, so that's one improvement I can have with the script so that it automatically kind of records down that information. So now what happens is if the user wants to do that, they could choose this user annotation option, right? And then when they run the image, there's 3 buttons here where they can say, if they see a cell, they say, okay, yes, there is a cell. If there's no cell, they say no. Or if there's 2 cells where like they can't tell, they just say maybe, right? They say yes, and so on. And these information are recorded in an Excel sheet. And then so at the end, when it's done, it tells you total number of yeses you said was, let's say it's something like 70. And then you say, okay, 70 is good enough. I'm going to run this chip, right? So at the end of the day, the other feature of this software that I recently added is the ability to generate a mosaic of images. So we use some other script. So basically, each experiment is defined by a date, a name, and the chip ID, which is unique to the chip. And all the images are saved in separate capture sites. That's the fluorescent images and the phase image. And at the end of the day, it generates this mosaic image, which is all 96 images with fluorescent and phase overlaid on top of each other in the well-play format. So it turns out that each capture site does not always correspond to the same well, right? So this is basically useful for selecting which cell goes into the library prep, which cell does it. And you can use the colors to later correlate, okay, let's say this chamber had 2 cells, this one had 2 cells. For example, some of the cells are dead. For example, this one looks like it's dying and so on. So basically that's what the user interface does. And
you take these at 10x or 20x?
This is 20x. You can also take it with 10x, and that will include the little number that labels, oh, this capture site is capture site 40.
Is that phase, or is there some
It's overlaid.
standard-
It's overlaid. So this is actually 3 channels overlaid on each other. So this is phase, and then it has the green and it has the YFP, the GFP and the YFP.
Oh, okay, so these were all YFP positive cells? Or?
These are all GFP positive, and some of them are YFP positive. Yeah, and the GFP is the live stain, the YFP is the dead stain.
You can just manually crank up the fluorescence up and down.
Yes, yes. And another way people use this is they could use different colors to label different cell types in fact sorting. And they just stain a little brighter, that way they can also see it on the image. And they use like DAPI to label live dead cells. Then you have 3 channels, and you can actually, in the same chips, sequence 2 populations and later correlate which one's which.
So, is that software going to become available for?
I can try to make it available.
Why don't you do a manuscript on it and
sure.
just publish the,
yeah. So I'm done with my talk. Yeah. So yeah. For example, this has two different colors and a DAPI to label if it's dead, right? And the 2 different colors is actually 2 different cell type. This is from a person's experiment. Right, so this way, like we only have 1 machine. Oh, let's say you only have 1 machine, then you can do 2 cell types on the same machine, right? The same chip. Yeah.
fantastic job.
Thanks.
No, we definitely, everybody needs to be able to use
it.
that. That is so cool.
Yeah, so I actually have an agreement with Leica, so they gave me the method of communicating with their scope, right? So I'm talking directly.
The code to move
yes,
to
move the stage and move the change the objective and so on, right?
Yeah, they should just license your software and then incorporate it into them.
Yeah, okay, all right.
Did you
rotate?
Yeah, okay.
The digestion steps, these lysis steps, can be pretty harsh on the cells. So resuspending the cells in a buffer, kind of keeping them happy and viable is a good idea. Typically, I'll use a media-based DMIM F12 is one example, and others are PMI and adding serum. I mean, again, considerations, do you be working in serum-free conditions? Is there an animal offer that you guys could use? In my case, it's not as much of an issue. So getting some serum is a good idea, maintain viability. Also, you want to disrupt as much as possible cell aggregation of the cells. For me, it's been a main issue with epithelial cells especially. So to do this, we typically use a buffer that has a little bit of BSA, so bovine serum albumin, as well as a chelator to ETA. So remember, we were talking about using that to wash off macrophages, but it's also good at disrupting the cell-cell junctions between epithelial cells. So my buffer, when I'm moving on from separating out my single-cell suspension to sorting cells, will be a basis of BSA and EDTA. In this case, and PBS, phosphorythanoate. So again, you can play with these things. If you feel like you need the media, you can probably do this possibly in some of these sessions. These are things that can be quite manageable.
They're very interesting.
Okay, so we talked about this a little bit already. So max versus fax, using kind of the simple tabletop magnetic bead kind of strategy. Positive is kind of obvious. It's super easy. It's super simple. And so it's ideal if you you kind of have digital selection using your markers. Like either they have marker X, Y, and Z, they don't have markers A, B, and C, it's on or off. This works pretty well for that. So in my case, I have the benefit of kind of doing most of my stuff using magnetic bead sorter. It's also considered less harsh. You're not pumping them through. You have fax sorter pushing them through a very small nozzle. Some cells.
Yeah, so the cells are moved through the glass pipette, and then the actual lysis or PCR
also time. Can you get on the sorter that day? So it's kind of more pragmatic. Fax is ideal, obviously, as you probably know, for rare populations where you're going to start with this, millions of cells getting down to that 0.2% population. It's also the case where your marker discrimination isn't necessarily just digital, but it's just this receptor positivity is like high, medium, low. You can actually detect with higher resolution.
Yeah, that is nice, but you don't benefit from this.
So clearly, you're going to, in most cases, have to be dealing with that over rare populations, I would say, especially when you get to the university. Oh man, that'd be a great question for such laboratories as Weissman, as well as the Hirschfeld group downstairs. So back in the Beckman facility, we have the fax core, who's the group who developed the fax technology. And so I've seen literature talking about 0.2, 0.02. I think it's depending on how many cells you have to start with. In the case of RNA-seq, as long as you can hit 2,000 cells to load.
Reagents as well as the cells. This will go into basically a really fancy.
That's true. That's right. That's exactly.
Which, in addition to being a thermocycler, also
though
has
I would still counter to you and say if you would be okay with let's say 60% your cell of interest, 40% a very similar cell type, you will probably gain in the analysis by having that comparison built into the sequencing data.
100% yes.
You've loaded it the same, you've isolated the cDNA the same, done all the steps for transcription, codification, all the stuff they're going to talk about, so I'll stop talking about it the exact same way. So you can really trust that it's like an apples to apples comparison. So that would be my caveat. And I've my terms only benefit from having
if you're just loading cells, that takes half.
So keep that in mind. So you can go apparently quite low. It's just whether or not that 0.0% equals 2,000 cells. I guess is the answer.
Good evening.
But
so I'll try not to be too repetitive or boring as to what you guys already know about cell isolation and purification. But hopefully there's some new resources in here that you guys can kind of look at and kind of gain from. My background is in developmental biology. My previous life was in cell biology, and I'm in the Krasnow laboratory studying lung development and lung disease. All right, so I can't help but have this slide in here talking about my perspective as a developmental biologist or a cell biologist, why I think this technology is useful. I'm sure you've heard all sorts of flowery exposition of the glories that is single-cell RNA sequencing. But so let's just kind of step through these. The first one's fairly straightforward. You suspect cellular heterogeneity from prior experiment. Example specifically being you do a culture assay, vary something, and you see a phenotype with some variance of penetrance, not complete penetrance. It's repeatable. You think, okay, there's probably some basis to this. Perhaps it's transcriptional fate related. So that's an obvious reason to do. And you don't have markers to distinguish between those two guys or populations. It's a good example to use it. One that I arrived at for its utility, and maybe a lot of you guys are interested in, is separating out or seeing populations of interest without any known distinguishing markers between them. In my case, I was trying to look at this progenitor in the lung versus two cells that it gives rise to, and I didn't have three independent markers being able to purify them separately. I attempted the strategy in microarray and doing comparisons, and I wasn't really backing out the program that I needed to get at what's the mechanism of this fate changing. So this method was really useful, and to some extent unbiasedly identifying different patterns of genes that are turning on these different populations and distinguish them. And perhaps novel markers can be pulled out from this data. And the one that I'm especially biased towards as a developmental biologist is deconvoluting, time course studies, and development is the best time course study. And from this, you're not just getting the transcriptional profiles of the progenitor of the differentiated cell types, but you're getting the continuum, you're getting the intermediates that are at different stages differentiating to either side, which I think is the real power and the real uniqueness of this technique. And I think it helped a lot of fields kind of establish the truth of these different programs and letting their differentiation and whatnot. Okay, So what are the main considerations? So your floor, or the minimum number of cells that you need to load onto the chip, is around 2,000 to 5,000, which compared to other assays you've probably used in the past isn't too bad. So, maybe tougher for rare populations, but that's the number you need to hit. Viability, as always, is critical, and obviously can affect your transcriptional, the quality of the transcriptional profiling you can get from this. And so it's good to kind of hit that sweet spot between getting a high number of cells, but getting, usable viability of the cells. So also keep in mind the impact of isolation itself on, or the potential impact rather, on transcriptional profiles that you'll see. It's been argued that perhaps the injury response from isolating these cells could affect transcriptional profiles. Heat shock proteins have been kind of brought up in past groups, so it's something to just keep in mind of. And it just means go as fast as possible, do as many steps at 4 degrees as possible, just to minimize that potential impact, because you don't want that to obscure the transcriptional profiles you actually care about. Okay, and this is one I like to harp about, the degree of heterogeneity of the population at time purification, at time of loading onto the chip. I think classically what we tend to want to do is to isolate all these populations from each other and do comparisons. And then I kind of think that the real positive of this technology is to kind of take a step back and cast a wider net, purposefully grab different populations, and even one that is known to be distinctly different transcriptionally and even of a distinct lineage. So in this case, I'm indicating the airway epithelium versus the alveolar epithelium, which differentiates from each other much earlier. So having that in the data set and seeing what's a known and significant difference versus allowed to give us a kind of a degree of difference, a gauge of difference, then compare type 2 to type 1, bipotent to type 1, so on and so forth. All right, here's the pipeline that we're talking about. This is the pipeline that you guys are seeing ad nauseum. Here's the part that we're covering here. Dissection, speed, sterility, if you can use under the hood, great. Here's the rig that I classically use to do my isolations on adult lungs for lung epithelium, and this kind of going into the trachea. Here I can do on the isolation of fluids. Okay, so you set up your rig nicely, that's great. If you can do that under the hood, fantastic, but sometimes it's not feasible. If you can remove any undesired cell types from your isolation prior to digestion, that's great. For example, we do a perfusion of the lung vasculature to remove blood lineages. This is typically an injection of PBS or media through the right ventricle back into the lungs. Another thing we do using this strategy here is the installation or lavaging of fluid into the lung itself with PBS and ETA. This is softening the adhesions of the macrophages and other cell types that are just sticking onto the lumen that I don't want. So if you can do a strategy like this with your tissues or solid tissues, it's recommended. All right, for solid tissues, digestion is obviously critical. So strategic choice of enzyme type, enzyme strength, a fantastic resource besides the literature of whatever field you're in is Worthington Biochemistry. They have a great run through of the different enzymes, the different strengths, different specificities, as well as amassed worksheets of all the protocols that have been used in the past on different tissues, different organisms, how they fared. So if you haven't checked that out, I would suggest you do, as it's been really useful for my work. Context of digestion. So you either mince the tissue, digest it, or you could be doing it intact. So in this case, I'm actually instilling the digestion fluid, from the trachea into the lobes of the adult lung. This is allowing me to biasly digest the cells from the tissue, and allows me to enrich without even doing any purification of epithelial cells. So if you use a strategy like that, it's pretty useful. Here's an example of a table based on digestive strength of the different enzymes. You have trypsin at the top, as you might guess, elastase, the various collagenases. Also considerations of like animal origin or not, maybe for stem cells that matters. If you're worried about bovine encephaly, spongivorm encephaly, it's good to have that in mind, choose your enzymes, great. So you've digested. Next step is to actually separate the cells from the non-cellular components. Either if you've minced, that's great, you're going to mince subsequent after digestion. Triturate, which is a fancy word for just pipetting up and down and breaking apart the tissue in smaller bits. Filtration, BD Biosciences will provide these 100 micron mesh filters or 40 micron mesh filters that fit into 50 ml Falcon tubes. So that's a nice way of removing the tissue from it. Next up would be lysing red blood cells from your cell suspension. So you're typically out of fluorescence, so for FACS or other purposes, it's kind of an issue. BD provides a kit to do this step, to lyse red blood cells. It's, I think, like an ammonium salt concentration thing, but check me on that. So you can also remove dead cells and small debris through such strategies as Percol gradients, which is based pretty much on density, which is kind of an old guard strategy, but still is pretty useful, especially for neurons or other cell types where different sizes, different shapes would affect where they would resolve in the gradient. So it's kind of tougher. You have to do your grunt work figuring out where your cells are in that gradient, but people report very positive outcomes with that. Maintaining the viability of cells after all this process, we're suspending them for subsequent purification. Keeping them in a media formulation, adding some serum. If you can add serum in your assay is a good idea. Trying to promote them to be single cells and not aggregates of cells. That's adding BSA, EDTA, or doing subsequent filtration through the meshes helps achieve that. Max is kind of a simple, straightforward strategy here. You can do it at the tabletop. If you have digital markers for positivity and negativity of the cells, I suggest you use it. It's less harsh on the cells. Fax is ideal for doing, sort of viability, size, singularity. The markers can be analog. It can be receptor high, medium, or low. So those are the different options you have before you. Quality control. It sounds like a joke, but definitely check your cells if you're running this expensive experiment. Double check marker positivity. People have done it before, so that's why I'm saying it. So there you go.
No.
Thanks.
The only reagent that you're using more of is the cell.
If you guys have any questions, feel free to email me or send me a power.
So you can test with your finger. Once you open it, the only thing you can do is basically put your chip in and load it. Now, so this is a place that you can actually mess things up, but this is really the only thing you can mess things up. So as long as you don't put it upside down or barcode facing the wrong way, then I think you're going to be fine. So there's a marked corner here, there's a marked corner here. Voila, now it's loaded. If you close, it will not pressurize, which means that you can close it without having a chip on. If you load, it will try to pressurize, and if there is no chip, it will basically give you an error message. It will say insufficient vacuum. Scan it right here. Right about now. So yep, there you are. Because so this is a pressurization pad. It's trying to pressurize. If there is no surface on top, it won't pressurize. Okay. Now, when you load the chip, every chip is barcoded, and there are barcodes that basically separate 3 different sizes of chips and 3 different chip types. There are chips for RNA-seq, DNA-seq, and gene expression analysis using Sagman probes or any PCR-based probe. And then for every one of these, there are 3 different sizes: small, medium, and large. Small is 5 to 10 microns. Medium is 10 to 15 or 17, I'm not sure, and the large one is 17 to 25 microns. Here, once you load the chip, it will read the barcode, and it will give you only the protocols that correspond to that barcode. So you cannot run a DNA-seq protocol on an RNA-seq. Well, actually, maybe you can.
So can you tell from this is RNA-seq or DNA?
Yeah, so basically I didn't show you, but there is a barcode here, and the barcode basically here is different depending on if it's an RNA, DNA, STA, medium, small, and large.
Just the barcode or the format is the same?
The format is exactly the same. I mean, you won't be able to tell the chips apart if you just look at them, but the barcode basically tells you what kind of chip it is. Now, there are 5 protocols you can run. From Fluidigm, you can also make your own custom ones, as Brian showed you. So what you can do is that first you can prime, and priming fills up the control lanes so that you can actually control the flow. Then you can load your cells, or you can load your cells and stain them. When you load your cells, basically they will go in and get captured, and then if you want to stain them for viability or some surface marker, God knows what, you just add that reagent here, and it will take for the medium and the large chip half an hour more, so total 1 hour to load the chips and stain them. You can also pre-stain your cells and just load them yourselves in half an hour. Now, once you're done with that, you take out the chip, and this is the moment of truth. You go to the microscope, you beam the microscope, yes, and then you actually image the chip and see what you get. If you didn't get anything, or very little, I know Fluidigm doesn't recommend that, but I feel you can reload the chip, so you can use the same cells, maybe at a higher concentration, and then try a second load just in case you capture anything more. Once you're done loading, then basically you put the chip back, you add the lysis, RT, and PCR reagents, and then the machine will just do sequentially the lysis, the RT, the PCR, and then you tell it when you want to harvest. Usually, if you run in the afternoon, it takes 8 hours to do the whole protocol. You don't want to come in the middle of the night, so you say, well, harvest at 10 o'clock. So you come back at 10 o'clock, and all the harvest positions, which are basically the ones here and here, will be filled with cDNA. You type it out, and then you go to the next workstation to see what kind of quality we get. And that's more or less how it works. It's not nuclear physics, it's very easy. Do you have any questions?
Gel, and then electrophoresis your sample through and takes an image in here. And then this is a part of how we are analyzing the samples, positive or negative. So you get the images off of the microscope, and you can kind of assess whether or not there's
Gary, can you wrap up and rotate?
No, they just changed.
Oh, you already rotated. Great.
So often we see that there's a single cell or multiple cells or no cell, and we get a different result over here. And so that is something to take into consideration.
Trey moved on.
when you're deciding whether or not to do a library. We're working on building a database that crawls through this computer every night. Ben, who's around here somewhere, is making it in Django, and so it actually will go through and find, so this one isn't named right, but on the chips there's a barcode, and so if you name each one correctly so it has the barcode and the cell number, later on the database will crawl through and it'll assign the image from the microscope, the image from here, the concentrations. There's a bunch of other information about size distribution,
The RNA-seq is basically compatible to work with
the average size, concentration, what percent is in the range that we're analyzing, which is this upper range over here. And so that can be fed in downstream to the library prep. I'm hoping that we get this. Okay. It's a mosquito. It's a got it. Good. Liquid handling robot handles small volumes that we could just feed in the concentrations, so it'll just do all the dilutions automatically. They're all different, but it would be very uniform library preps and completely hands-off.
And there are ways, so the machine will not let you do this.
So if you look up here at the gel-like image, you can see these two peaks in here. These are the RNA spike-ins.
Yes.
Oh,
ERCC
the machine
from
will basically
Ambion.
read this as soon as you insert it. 177
They just kind of show that the chemistry is working.
So if you want to do,
you add these in, and it's kind of a negative control.
you either need to print your own barcodes or you
Those go into, it's just like.
It goes into your
part,
lysis
Okay,
solution.
okay. So that's just part of the reaction.
Right.
Okay.
So one of the important things is that if you have a cell that doesn't have much RNA in it, like T cells, you need to dilute these like a millionfold further than maybe standard protocol is.
So are those sequences in there when you go and make your libraries? So you can, okay, so that's something, we'll do that in my analysis then, is that you can use that. I didn't realize that that was a passive reference in every single sequence. Okay, it's good to know.
There's a reference genome for them too.
Okay. Alright, this is like ERCC's mic in. Try and use that.
So qualitatively, you're looking for this band up here in the larger fragment size. That's where a healthy and viable cell will have its transcripts.
With this corresponds.
That's
So you know by reference all the stuff was just spiking. I didn't realize this. I thought it was just like one well and had it, but it's in every single well.
So basically you see only one
I can try and then
one uniform
samples. So we can actually like see how it correlates with like right? Because you're supposed to get
So the
like,
bands
like
aren't
expected
the cDNA really. So this one up here is the upper marker, and this one down here, these two bright ones are markers. This one down here is a primer. These two are the spike ends, but then the smear is the cDNA. And generally it's up here in the 1 to 3 kV range. I don't know if that answers your question.
You know what the limit? So we're working with small plasma cells, they don't have much RNA. So when we do the fragment analysis, we never really see like a broad peak of that. But of course,
So you see something hopefully better than
yeah,
these
better
guys?
than that, but
it's more like a little bump.
But
okay,
like,
so these are the ERCC.
yeah, that's kind of stuff.
Yeah, we see stuff above that.
Okay.
But
So do we look at like fluorescent intensity units? We look at concentration. Like what is a good like determinant of like what's a good cell to sequence?
Those numbers are going to be very skewed. If you do have these two peaks, that's going to be like 100% of the value coming out of this one.
So just looking at the peaks themselves rather than just this one.
Qualitatively, yes, but I think for making the library, you'd have to use the concentration, including ERCC.
So you could probably look at like the ratio of the size of those peaks compared to the entire peak within there, right? As your reference to see like how big your concentration is.
You can actually see that there is
We've made a custom script that does more amplification. You may want to
I'm Jeff. Let's see, Jeff Stanley. I'm a second year in the Quake Lab, joined recently in the biophysics program here at Stanford. And I'd like to talk about quality control of your RNA-seq data. This is mostly sort of after alignment and understanding, when you have a good cell, or a good sample, and when you have a poor sample. And when you might want to exclude that from from sort of further analysis of the data. And I'm going to make this fairly short because quality control, I think, of your samples and determining what is a good cell, what's a useful cell, really depends on your application, and depends on the cell types you're looking at. Depends on the tissues you're taking from, whether they're fresh tissues, whether they require long dissociation protocols, whether it's frozen cells that have been reconstituted. There's going to be big variations in quality, and so how stringent you want your quality control is going to depend a lot on the particulars of your data sets. So generally, what causes low quality single cell data? Well, the trivial case would be no captured single cell. You can have your cell might not be fully lysed. Maybe this might be a problem if you're using fixed cells or something like that. That can be a big problem. A big one is poor cell health or RNA degradation. This might be due to a long dissociation protocol. You're taking a tissue and you're putting it through a lot of stress. Or you have samples that have been frozen down and you're reconstituting them, and you have cells that have been stressed that way and are dying, or their membranes have been ruptured and they've lost RNAs, things like that. And then there's obviously the possibility of failure of the biochemical amplification step in the C1 chip, and that's pretty rare. I think that's quite uncommon.
It's probably very common, but it occurs at a consistent level.
Really? Okay.
Yeah. I don't think that we have a good number for the efficiency rate of mRNA to
yeah.
cDNA conversion.
Yeah, I mean, yeah, right. Okay, so yeah, there's a lot of noise in the data. That's sort of known, but in terms of random dropout and complete failure of a reaction is pretty rare. So yeah, there's a lot of noise, a lot of dropout, but it's consistent cell to cell.
In that you're you parenthetically said that you're dealing with some fixed cells and then having variable release of them.
Yeah.
That's a really cool thing to be able to deal with partially fixed cells.
Mm-hmm. So
are you
that's.
going to say anything more about that? Or?
Yeah, I don't have so much experience with that. Actually, you might want to talk to Spiros later. He's worked with fixed. He's spent some time trying to get fixed cells to work. It's not so trivial. There's some issues. Yeah. I mean, it's very neat if you can do it, because then you have access to all these tissues, especially human tissues that have been preserved. Human samples from all kinds of interesting cases, but it's not trivial. Okay, so the basic QC, I think earlier today you guys touched on this. So obviously you have your image. Here I have cells that I sorted and I stained them with DAPI in the media. So so DAPI is a chemical that binds to DNA and becomes fluorescent, but it is typically impermeable to the cell membrane. It can't cross through the cell membrane unless the cell is ruptured at some point and takes in the DAPI from the environment. This is also sort of a signal that the cell is maybe damaged, maybe also taking RNases in from the environment, and is potentially a marker of low cell health, low cell quality. And then here's a cell that was sorted. This is actually from a transgenic animal. It's endogenous fluorescence, and you can see that it's it doesn't really show any DAPI, so it probably didn't rupture much during the various dissociation and loading protocols, and it's these fluorescent proteins that it's been expressing are still there, still present. So unlike this cell, which lost its fluorescent proteins that it was expressing and allowed DAPI into it. So then you look at the output of the fragment analyzer and the cDNA that you get after amplifying these. Obviously with no cell you get no cDNA. With a good cell like this that's healthy, you get cDNA that's where nearly all of the amplified transcripts are between 450 and 4,500 base pairs in size. But you can see for a dying cell like this, a stressed cell like this, there's a large fraction of the reads that are between 100 and 450 base pairs. So these are presumably degraded RNA that's in the cell. It's present. It's more degraded than the healthy cell for whatever reason, and not quite as many amplicons that are the normal length that you might expect. And so the fragment analyzer quantifies the average size of your cDNA as well as the quantity. So you generally get your samples on the quantity, and the average size gives you some idea of whether it's degraded. Here it's 1,100 base pairs average size. For this real nice cell, the average size is more like 1,700 base pairs. However, I'd still always recommend sequencing both of these samples that have cDNA and have some sort of cell in them, because you don't really know in your downstream analysis how this degradation is going to affect your results, and I'll talk a little more about that. So basic quality control of the alignment, you want to a good cell that produced plenty of cDNA and where your library preparation worked well, will should have a number of reads that are mapped. That's roughly the sequencer output divided by the number of samples you put in the sequencer. So a NextSeq might give you 130 million reads total. So if you put in 96 single cells, you might expect 1 or 2 million reads per cell, and you see that here. You have 2.5 million reads in this cell, and 90% of them were able to be mapped to the genome. Here in this cell that's much lower quality, you only have 40,000 reads total, and only 40% of those were actually able to be mapped to the genome. So clearly something went wrong here, whether it was in the C1 amplification or the library preparation or just the cell was very low quality and contained little RNA. So this, Ben briefly mentioned this package RNA-seq. So this is sort of quality control after the alignment, after doing top hat, and you've mapped your reads to the genome. One thing that's useful to look at is the gene coverage. So RNA quality tends to correlate with 3' bias. The clone tech amplification is, as I think this has been talked about, is poly A primed, and so if you have degradation, you're going to amplify just the 3' end and whatever's left over after having been degraded. And so more degraded samples with RNA that's been fragmented are going to have a lot more 3' bias, and you can see that in here. So here's a good sample from the previous slide, and the coverage is really not bad at all. There's some 3' bias, which is I think unavoidable when you're doing the single cell and you're doing these poly A tailed amplification. But you can see in a degraded cell, you have a lot of 3', you have very heavy 3' bias. This is another, so this is the gene body coverage Python script from RNA-seq. You can also look at the percent, this is not high resolution enough here, but you can look at the percent of novel junctions mapped in top hat. So top hat will search, in addition to known splice junctions, it'll search for novel splice junctions that are not in your reference genome or reference annotation. And so this junction annotation script from RNA-seq will plot the here in green and red we have the percent of junctions that are novel. And so you can see in this degraded sample, if you can see, this bit of the pie chart is actually higher. So you actually have a higher percentage of novel exons in the more degraded sample. Now that's probably not because the more degraded sample is also somehow has more interesting junctions in it just natively, but rather that the degradation has caused cutting of the transcripts at certain places where they normally wouldn't be cut, and causing sort of an artificial junction that top hat calls. And so the typically in good quality cells there's around 2 to 3% novel junction that top hat calls, but in the lower quality cells you may have 6 to 10% novel junctions. Then here, this is another read distribution, is another Python script from RNA-seq, and just shows you, if you have the proper annotated file, where your reads, what the coverage is of different gene features. So introns, 3' untranslated regions, 5' untranslated regions, and then the actual coding sequence exons. And you can see in the higher quality data set, the coding sequence has a higher relative coverage than in the degraded sample, and the degraded sample has a higher coverage of the 3' UTR, which suggests that it has that there's more degradation because the 3' UTR is right next to the poly A tail. And this has implications, I think, not so much for raw expression values, but for looking for things like splice variants. So when you look for splice variants, you're looking towards reads that have been mapped to the actual exons. And so if you have a low quality, lower quality, more degraded RNA like this, you have less reads you have less coverage of your exons. And you have potentially less ability to call splice variants and understand the diversity of splice variants in your single cells.
I would call this probably only moderately degraded, maybe slightly worse than moderately degraded.
Yeah.
But in terms of, like you were saying, read per gene, or kind of estimation of gene expression level in that cell, it's probably you can come fairly close to estimating it as it would have been in a healthy state.
Right, for gene expression.
Yeah.
Right, yeah. So I do think that the gene expression U10 is the estimation is not too off when you have 3' degradation. What I'll show later is that I think there's more dropout. There's a higher dropout rate of genes when you have a degraded sample. You also might assume that if you're using that longer transcripts are going to be relatively less represented if you use an FPKM that's normalized the transcript length. But yeah, I think that there is higher dropout in these samples. Partly there's just tends to be less cDNA produced because the RNA is.
What percentage of cells would you say in a generally healthy study are showing this kind of a pattern?
Oh, it really quite depends on the tissue, on the so for the stuff I've been doing, which is mouse tissue, and a fairly lengthy dissociation protocol. It can be between 10 and 50% of the cells are sort of ruptured, they're DAPI positive, and they're degraded, and it depends a lot on how well you handle. Yeah. Okay. So I'm being told to hurry up. Okay. I'm sorry.
Can I ask just a quick? So
Yeah.
I tried to use that package in the past, and it's
Yeah.
a really big pain in the butt because you have
Really?
to run all these different steps. I don't know if it's changed at all, but is there something more streamlined where you can just feed it the BAM file without having to do all the extra? Because I know you have to add all these extra annotations before you run it.
Well, yeah. I mean, so what I just use is you just use like a GTF file, right? Which you can get from various sources. I mean, it's the same GTF file you might use for your alignment.
So you no longer have to do all the different, because I remember there were like 5 different extra annotation steps to like the actual BAM
Could you send out a
sequence
link
itself.
for your code and as a supplemental link later?
Yeah. I'll send out the code I've used for all these things. But yeah. It wasn't too hard for me, so maybe it's updated. Okay. So right. So then quality control. Once we get into expression values, I think is a valuable way of doing quality control. So housekeeping genes are genes that should sort of be expressed in broadly in cells and are used for important metabolic processes or structural processes across a lot of different cell types. So we used a set of housekeeping genes, including beta-actin, GAPDH, heat shock protein 90, so the interesting thing to notice is that basically most of these housekeeping genes are not that great markers. There's a lot of dropout of most of them, except for beta-actin. So for cells that don't express much beta-actin, they don't express much of any gene. So these are clearly quite low quality cells.
GAPDH is surprisingly good too, actually. I think.
Really? Yeah. I think there's a fair amount of dropout of it. I think if you have a more stringent quality control, then you might consider gating on GAPDH as well. But as you can see, there's sort of a lot of dropout of GAPDH, even in places where there's plenty of beta-actin expression and heat shock protein 90 expression, among other things. But I'll show. So this requires a couple R tools, hclust heatmap.2 to generate this plot, and then cut tree is something that where you can cut this tree and specifically pick out these cells that cluster here and that have overall low expression of your housekeeping genes.
I have a quick question.
Yeah.
So, in the first part of today's session, they said you can pick up as low as a single molecule from the cell sample of interest. And these, presumably, there's many RNAs of ACG and et cetera, right?
Yeah.
So do you think it's a matter of that cell just didn't actually have that, or somewhere it actually got lost?
So I think these tend to be cells. So you can see there's actually very low gene expression of beta-actin. It's hard to tell because of the way this is set, but there are going to be some fragments of beta-actin here, but these are cells that overall have didn't have many reads mapped to the genome in the first place, I think, and had just overall very small amount of cDNA.
So I think it was a starting input RNA that was the issue, not like you lost beta-actin.
Right. It's generally the cell itself is very low quality for some reason. And then
hundreds of FPKM for a active read.
Yeah. Beta-actin is normally expressed very high. This is 2 to the 10, so up to like 1,000 FPKM. And then this is a similar plot with a smaller set of cells, and you can see that all of these cells that I have do express beta-actin. It's a little hard to tell with the color balance here, but this set of cells seems to cluster on generally expressing beta-actin lower, and they have a lot more dropout of these other housekeeping genes that we have. And so these are, as I mentioned earlier, these are samples that tend to be more degraded, tend to, if you stain for live dead, tend to have dead stain, tend to have been ruptured at some point during the process. Whereas these cells that express quite high amounts of beta-actin and don't have much dropout of these housekeeping genes tend to also have very, very good 3', 5' coverage. You can tell here that there's actually sort of a bimodal distribution of beta-actin expression, where you have this large population of cells that express it quite highly, and then this smaller population that express it somewhat more low. And I wouldn't say that you should automatically take these cells out of any analysis, but you should flag them. And when you're trying to do things like phenotype detection and detecting subpopulations, if you get a subpopulation that's entirely these cells, you might think, okay, maybe this isn't actually a real subpopulation. These are just sort of degraded cells and have a different expression pattern because of that. See, there was also an analysis done. This is Aviv Regev's paper. They had quite a lot of cells, and they sort of trained a support vector machine, by looking at actually wells where there were no cell, and correlating, doing a sort of correlation measure of all their cells to the no well, no tube control, and sort of generating, in an unbiased way, a failure score that was basically how similar this cell was to an empty well. And they actually they removed almost like half their samples or a third of their samples. And I think this, again, sort of depends on what you want to do with your analysis. If you're just looking to call a phenotype, a degraded cell is not going to, you're probably going to be able to see the phenotype, especially if you have some marker genes you're looking for, as long as they're highly expressed.
That phenotype is revealed over thousands of genes. So you can, once you see the distribution in terms of recount into those really bad cells, it's very easy to get them classified.
Yeah. Right. But then if you're trying to say some more subtle things, and so I think in this paper they're trying to say some pretty subtle things about the statistical distribution of gene expression, so things about the mean and things about the variance of gene expression. And so I think they try to have a very tight quality control measure for that, because now you're getting into to much more subtle measures of gene expression that can be affected by low cell health. So you might see some bimodal distribution that's not really a result of real differences in phenotype or something like that. And then so Fluidigm singular also has a pretty useful quality control method, where they basically take the genes, take all the cells and find the genes that are expressed in at least half the cells, and then do a PCA on those sort of constitutive genes that are discovered in an unbiased way, and you can that, and you do it. Yeah, so PCA. So this is principal component analysis. Actually hasn't been talked about yet, but this is basically to find the maximum variance in that data, and this is going to be variance in sort of these constitutive genes, and so you tend to see these low quality cells that have low overall expression cluster together, and so these are things that you can flag for downstream analysis. So contamination detection, this was sort of done by Spiros and Winston Co. And so in a lot of samples, you tend to get cells from from the blood, things like that, that you may not be interested in this particular tissue. So for example, a human brain or a human pancreas or something may have a lot of blood cells flowing through it. You're not so interested in phenotyping the blood cells that are passing through a human brain because they're going to be pretty similar throughout the body. So this GTEX, which is this database of, I think, something like 1,800 samples and a lot of different tissue types. And so what you can do is correlate your single cell expression distribution, single cell expression pattern to these bulk measures of tissues. And then what they do is actually use a hypergeometric test to derive sort of a p-value for what is this cell, what tissue type is this cell most similar to. And so you can sort of pull out, these are two cells from a brain tissue experiment that Spiros did. And so this cell correlates quite well to bulk expression measurements of the brain. So it's likely a brain cell. But this cell from this brain dissociation experiment actually correlates very well to blood tissue samples and not particularly well to brain samples. So there's a very good chance that this is a blood cell that made its way into the sample. So this is just a resource. This is the websites here, gtxportal.org. And I think this code could be. So this is Galaxy. This was mentioned already. It's at usegalaxy.org. I was just going to talk about it briefly because it's a way to quickly get started, like you guys said, but I think we already discussed that. So any questions? You good? Okay.
So I was told I have 10 minutes before you guys leave, so I have 10 minutes to tell you how to make your data work for you. And one of the main things I'll go in is that every paper that you read, you see these plots. So you see basically they say I perform the principal component analysis. I show you the plot, I show you these different cells, they're all grouped together very nicely, and I show you a bunch of arrows. So my aim here is to let you guys finally understand or get a very quick mathematical intuition behind what PCA is, what the plot is actually telling you, what can you infer from this, and when does PCA breaks? When does it not apply? And when do you need more things to do? And finally, if you have time, I'll talk about some of the projects I'm working on that I'm personally interested in. So first thing, principal component analysis. That's where I brought this thing here. So in my notes I wrote here, I will tell you that the principal component analysis, it maximizes the variance of the projected data. It minimizes the distance between the individual and the projection. And it is one of the best representation of the diversity/variability of data. But what does that actually mean in real life? So a lot of times you have a data that's described by many, many different kinds of properties. Let's think about a solid object. If you have a point in space, it's probably you can describe it in three terms, x, y, and z. That's a point in space, x, y, and z. So in this case, let's say every object in this world can be described by the length that the object has, the height that object has, and the width that object has. And we say that is our measurement. And then we can put a plot. We can put a point here and say that maybe I'm this tall, I'm this wide, and then I have this. Yeah, luckily I'm not so much this way, but yeah. So each one object can be represented by features, basically the length, the height, and the width. And then we can plot it here. So in the end, if in this three-dimensional space, if we plot every object that we have here, we have this cloud of data that's here. And what happens is that this is only three things that you're measuring, three features you're measuring. Now imagine your single cell data. After all the hard work that you put through, you're measuring 30,000 genes. You're never going to be able to plot something like that. You want to put your single cell in that point, as a point on that plot, and yet to be able to project the image such that you actually make sense out of it. So what PCA does is precisely that. this is the actual data. What it does is that it tries to find the direction that has the largest variance, which we are going to say the largest variance is the ones that hold the most interesting information. And then we are going to tilt the thing such that we are able to see the clustering of the cells. So what I have here is a conical flux. So I have 3 points, the 3 blue points and the 4 red points. If you just imagine things, like these are actually your single cells, right? They are characterized in space. So if I show you the cells in this manner, if you look onward in this manner, all the points are all clustered together. You don't really see that they separate out from themselves. You do not know whether they are different cell types or they are different cell clusters. They are all smashed up together. But now if I were to tilt this axis, right? If I look at it from this perspective onwards, if I were to change the projection by which you see it, now you can see that there are actually 2 distinct clusters. So in 3-dimensional space, it's fine. You can get an object, I can rotate, turn, look at it, and I have a complete picture about how the system actually looks like. But when you have 30,000 dimensions, you're screwed. So what you do is that you say, please show me the first and the second, which best describe my data. Okay, so that's the intuition that I want to give you in 5 minutes. Normally I take a longer time over a cup of tea, but that's the intuition. So conceptually, in our mind, how do we think about this? So precisely at the end of the day, be it any software that you use, with all the quality control, you have a set of cells and you have a number of genes. These are the set of cells and the number of genes with high quality numbers or counts that you have that you believe is true. And now you want to ask, I did so much hard work, I collected maybe 200 cells. Is there any heterogeneity in it? Do they segregate differently? Of course, a lot of us can say we have our own favorite genes. We like to just look at it and compare across. But let's say if you take the entire transcriptome as a whole, without any supervised methods of actually telling the algorithm and everything, can we just separate them out? So precisely that's what PCA here will do. And that's what the first figure of the paper does. And then what you can see here is that this is a gene space, all right? So each cell is described by, let's say, 30,000 genes. And then each of them have a value. And you use that and plot a point in that space. So when I try to draw, it's very difficult to draw 30,000. So just imagine I draw 2, and then there are multiple other axes. And then the cells are there. So what happens is that now, in our context, we try to put our axis now in the middle of this clump where all the cells are, and they say, find me the direction that has the most variance. And then I say, okay, so I find all these new coordinates, and each new coordinate is orthogonal to each other because we want them to be as independent as each other, so they're orthogonal. And then once I have these new coordinates, I replot them in this space. And then now, you can see that cells that were described by many components, now it reduces to as many components as we want. So what happens here is that I will tell you about this software. So one of my aim today is also to empower each and every one of you to do this analysis on your own. It's not that hard. Once you have the set of numbers, you can do it. It's not that hard. So what happens is that you start with, and to be able to enable you to interpret very quickly some biological insights into it. So once you get this matrix, all right, n by n data points, and then after going through this conceptual process, finding the coordinates, redirecting it, and projecting it in the direction where you can see the clustering, we reduce the dimensions that we have to look at. You can think of each coordinate. It's actually each coordinate actually contains some components of each of the individual genes. Okay. So now we say that this component maybe has certain genes contributing to it, and it segregates the cell. So this is the conceptual pipeline. When does it break? When does PCA doesn't apply? Suddenly I tell you about this wonderful thing. It seems to work fantastically. No, there's no such thing as a perfect thing in life. Raw data. If I have raw data whereby a gene, let's say a cell is described by 2 genes, gene X and gene Y, and then this is their expression pattern. You expect PCA to do this for you, right? It makes more sense to do this, to project it this way and look at it this way. But no, it doesn't. It does this. Why? That's a mystery, huh? But if there's something that can be described by other polar, this is actually polar coordinates, this is circle, and if you do PCA, if your data looks like this, you do PCA, what do you get? You get this back. So there is a fundamental assumption underlying PCA, which is that your data must sort of be a multivariate Gaussian. Okay, so that term sounds very, it's not friendly at all, but I just want you to know that PCA doesn't always work. It fails sometimes. For the data that we're working with, it works to a great extent, but there are some caveats to it as well. But in general, it works quite well for the first level, looking at the heterogeneity of the data. It works well. All right. So
do you have a version that uses ICA?
Do I have a version that uses ICA? Gosh, you are a guy who's in the business. Yeah, I actually have a version that does the independent component analysis. But that one is slightly trickier because the interpretation of the S matrix, I mean, you have the mixing matrix and you have the other matrix. The interpretation of that matrix is slightly messier. It's more fun to do, I agree with you. But I'll go on first. We can talk about it later. Yeah. So I'm going to tell you that with just 2 blocks of code, you can get this kind of data. You can see the heterogeneity with just 2 blocks of codes. And these codes are all available in the paper, in the supplementary information. So you can just download it and look at it in the first page, that will be in the references that you have. So what happens is that we can use this package. This is actually my own data. I did it 2 weeks ago. So what I did was I just projected all my cells onto the principal components. And then I can see that from different tissues that I do, I actually get a very different representation from it. And the other block of code that you will see in the supplementary information of that paper is how to plot it, right? You don't just want to segregate the clusters. Okay, you can do the experiments with 100 cells, you separate them out, they are very nice clusters, you think they are different cells. But on top of that, we are humans, we have intuition, right? Let's use our intuition. Let's say we say this cluster is due to one tissue source, or this cluster is due to a batch of experiments that I did something to it. Or let's say this batch of cells is actually liver cells. My intuition say they are liver cells, or they are progenitors.
Liver contamination.
Or liver contamination in that case, it doesn't matter. So what we can do is that we can use this, and then we can color the points according to our intuition. And we can see how good our intuition is. You know, does it segregate itself out as well, and everything else. So now that I reached this point, what is the next thing that we are most interested in? I can see the 100 cells are heterogeneous, they have different clusters, they have different populations.
Publication.
Publications. You are 100% right. We want publication. As to publish, we need genes, right? We need to tell a story about the genes. So we need to know what are the genes that's contributing to the differences, which is why accompanying the PCA plot, normally they put out a variable plot. They give you a plot with arrows like this, and they say, yeah, you know, this one is specific to one population. These are the genes that's contributing to one population. So given our previous intuition about PCA, what we have is that each component is actually sort of like has components of the different genes as well. So basically, you know, I have multiple genes, that space, I project into 3 space, and each of this new coordinate system have certain elements of certain genes. So what happens is that to help you visualize, we can think of it this way. So these are that circle that you see is this slice here. Each one of this vector is like a gene in the space that we do. So we did a rotation and everything. We center the data, and we rotated the data to show the maximum variance. So now, when the genes, all the different gene axes that used to represent it now they are all twisted in different ways to reflect the maximum variance. So what it shows here is the component of the genes that actually contribute to the individual principal component. So basically, if you say that the first principal component is the one that describes the most variance, then you will be able to tell which are the genes that are the one that's contributing to the PC one that is differentiating the 2 different clusters. So you want to have that list. So how do you get that list of genes to get your publication? So in the package that I have, just one line, put in the function, and you get everything out. I'm not going to go through the math of the eigenvectors and the loadings and everything, because I think a lot of times you want to be quick, right? You want to be fast to publish. So this basically tells you what is the correlation, how much of that gene is contributing to that component that actually separates out your cluster. So that's what it does, and gives you a p-value of how significant it is. Now, with this gene list, you can do wonders. You can do experiments, qPCR, and everything. But I like to go a step further. I want to investigate, does this list of genes actually mean anything, right? I mean, a lot of people will do gene ontology and everything. I personally have an interest in just the very well-curated gene sets that I think the Broad has. And because a lot of times if you work with cancer, you work with canonical pathways and everything, or transcription factors, this data actually proves that all you need to do is take the list of genes from that function, toss it in, and what you get is, it can tell you, it has everything to do with extracellular matrix interactions, which makes me very happy because that's what the cells are actually undergoing at that time. But then, every one of us here, we are humans. We are not computers. We have an intuition. We have a favorite gene list. We have a list of genes that we know that are sort of important. So how do we compute significance? I'm not going to go through this because it's all in there. But I'm saying that what we do is that we can also do the same things on your favorite gene list. This one is a ball in the urn model. I'm not going to go through it in consideration of time. So I'm going to go something that I'm personally interested in. So I've talked a lot about how do you dissect heterogeneity? How can you tell a tissue is heterogeneous? So what can you do with single cell data? My personal interest is not only about tissue heterogeneity, but also of clonal analysis. You want to be able to study clones. You don't just want to say that the tissue are different. You want to say one came from the other, or that particular clone is growing. So over here is are things that I'm interested in. One of the things that I'm working on, which is I think Florence mentioned at the start of the talk, which is on how do you measure DNA amplification and how do you measure mutations. So over here, I'm actually measuring mutations in single cells for cancer. In particular ALL. And I'm just going to quickly show you the result, which is what you can do to give you guys like some a vision of what can be done. If you measure the mutations in every single cell, whether the mutation is present or absent, you theoretically could build a lineage tree. So what we have here is actually every single patient with cancer, with ALL, you do single cell capture, you get the DNA, you see the mutations, you can get a complete outlook of what are the different clones that's existing in the cancer, how the clones, how big they are, how they are derived from each other, and at a single time point, at a single snapshot. I believe that will increase, give you guys some things to think about.
So you didn't have any normal cells in there, or those are the two cells in the middle are the normal cells?
Yeah, I see. If your eyes are sharp enough, you will see the normal cells, which is somewhere here.
Yeah, but each patient should have normal cells.
No, we enrich for the, But sometimes we don't, the enrichment doesn't work that well. So we see the non-normal cells as well. So yeah, sharp eyes. So now, we can get the inferred clonal profile as well. What are the mutations that are present? What are the ones that are constant with each other? And which are the ones that are probably just normal cells? Okay, with that, I think I did 10 minutes. I'm good. All right. I would love to take any thoughts, questions, or inspirations that you guys have.
Do you need a copy of the video?
No.
That was great. There's no way that was 10 minutes.
Is it? Done. I was aiming for 10, really. But yeah, I'm hoping to hear your thoughts actually on the work that's being done. Yeah. Because I know that I have the easiest time of all the speakers because I get to speak about the big ideas. Yeah. Not the things, but a lot of effort has to be put in by the time you reach this stage.
If you go back, try to go backwards from the end of the talk into more the middle of the talk, and then think about the lineage derivations of, kind of genomic states, the indeterminately differentiated state. So now try to backtrack up the tree to the likely precursor state.
Yes.
And then how can we find sort of a common
precursor of two different terminally differentiated states?
That is that I think is the $1 million question, actually. So that's why sometimes, I mean, it's about biological intuition. You need to find a good biological model to answer certain kind of questions. In that case, if you want to find the lineages, it's always easier to measure mutations and to infer it from the gene expression profile. I'm not going to say that it's impossible. I mean, we know that there are developmental genes like SOPs and everything, whereby their expression actually correlates very well with the developmental time span of things. But then a lot of this things is going to be harder to infer. I'm not saying impossible, just harder to infer. And even if I came up with an algorithm, people wouldn't like always believe it until you do a real assay to show that in the mouse as well.
Nobody knows what the best real assay is for that, actually.
Precisely.
Yeah. But
Yeah.
let's say that allelic exclusion occurs at some frequency, and that once you finally decide on, a gene to really make out of this cell, that it tends to exclude the other alleles expression.
Yes, you can
and
use
that you have a heterozygous animal, and so now you have a high frequency of like allele heterozygosity. Do you think that we could come up with a method to find out when we're like a primitive state that has biallelic expression and then a more different that's the same?
Yeah, but that you see, you're leveraging on a known biological phenomenon already. My quick answer is yes, but obviously that would be like another 6 months of work down just trying to make sure that the noise.
6 months would be worth doing.
Quick estimation, yes.
I have an easy question. So that Facto minor,
Yes.
is that just a package available?
It's a package available in R. I know it may be difficult, huh? Sorry.
Is that like through bio? Because there are a ton of these like biology related R.
Yeah, in the bioconductor package. Yeah, this is not really in bioconductor. This is just a generalized things. Yeah, because a lot of the times by the time we reach this stage, right, the hard work has been put in. You get just genes and numbers. Yeah, be it FPKM or counts per genes, whether you want to sum up the different isoforms and everything. You can try different things. And then in the end, it's just a matrix of numbers. And when you reach that stage whereby you have just numbers, you can use anything that you want. I have a personal preference for this because it is really used. Yeah, you just need a good intuition. You don't need to know the math. I'm not saying that you shouldn't know the math, but it helps when it's intuitive and easy. Okay. Yeah.
My second question was, so
Yes.
you mentioned briefly that if you have like your favorite gene list and you
Yes.
want to look at, so can you like can you say to PCA, put all these genes into principal component 1 and then figure out principal component 2 and then map everything, or?
So you see, that's where it gets murky.
Okay.
Like PCA, PCA is meant to be non-supervised. Like the ground state, how it is, let me find the differences between them. Whereas if you want to start directing the genes to look at, essentially you just say, let's do supervised clustering. Let's just take this list of genes that I'm interested in and let's just cluster them, And then that one Jeff mentioned, the H class, the heat map.2, and you can just cluster them and take a look at them. But in general, like you did so much work. You got a whole transcriptome. Might as well use it. Yeah.
So
Yes.
the expression gene by cell matrix usually very sparse. There's a lot of zeros there. And zero actually is a mixed population, mixed distribution between the true zero and the dropout. Do you think it's appropriate to use PCA on such a high sparse matrix?
And how could you help that
Excellent question. Excellent question. Okay, I'll let you continue as well. Yes.
Yeah, how could you sort of avoid the bias of like how strong zero is by some of your filtering strategies?
Yes. You guys are going down into all my secrets now.
Right.
Yeah. So it's true, and it's also partly the reason why I'm trying to use independent component analysis, because that one, you don't need to assume the Gaussianity of the data. And it's true that the RNA-seq, the expression, I'm losing people. It's okay.
Transportation at like 1:30, so.
Oh.
Yeah, we thought it ended at 1.
Yeah. So yes. So one of the tricks that we use is you can think of it as a mixture model between the ones that have the dropout, which is more like a Poisson, and the ones that's expressed, which is more like a negative binomial. So that's what I'm working on as well. Can you differentiate the genes into these two populations And then you can also ask a question whether the genes actually switch between these two states from the one that's likely to be a dropout state into the negative binomial distribution, which is more likely to be an expressed state. And then within genes that are always constitutively always expressed, do they actually show a different expression? So that's yes, correct.
A few months ago.
Yep. Huh? It's not from me. No. I wanted to do it, but no. But there is some subtle differences between that and what I'm trying to pursue, because they if I'm not wrong, that's the single cell differential expression using Bayesian methods paper, right? Yeah. That you're referring to? Yes. Yeah. So that's the one whereby they try to do that. I presented in lab meeting, I tried certain things, and yes, I'm trying to exploit that to precisely solve those questions that you want. And hopefully it's a simple and intuitive manner that allows people to make quick interpretations of the data. Yeah. So yes, those are very good questions. Yeah. So I'm saying that paper is one of the nicest one to read, and it's fairly interesting. Yeah.
Morning guys, I'm Joe. I'm a 4th year graduate student in Mark Krasnow's lab. And what I'm going to be talking about is the first step in the C1 single cell RNA-seq process once you already have cells in a tube in suspension. So this is one of these chips that I'm going to be talking about, and I'll pass it around. They're one-time use, so I don't feel bad about touching it with my hands and passing it around. But and then this is the machine that I'll be talking about, which it gets loaded into. Let's see, so with those chips, there's a lot of aspects of loading cells and running one of these chips that are generic, meaning it's the same every time you do it. But there are a few steps that are pretty critical to think about depending on the cell type that you're using and the number of cells that you have. So, for instance, the first thing to think about, and when you go on Fluidigm's website, you'll find that there are actually 3 different size options for chips that you can order: small, medium, and large. Small, they say, are for cells of diameter 5 to 10 microns, medium 10 to 17, and large 17 to 25. I can tell you in my experience with lung epithelial cells, the medium size chip works great. I know the hematopoietic people in Irv's lab typically use the smaller ones. And for the bigger chips, I don't know anyone personally who's been using them, but I could imagine, like, for cardiomyocytes or neurons, that might be the way to go, as long as those cells can stay alive. But if you think that your cell is so big that its diameter in suspension is larger than 25, or at the worst 30 microns, you may want to think about just manually picking cells and doing the protocol that way. So, independently of the chip. Okay, so let's say you roughly know the size of your cells, which gives you the information you need to order one of these. It'll arrive in the mail, and it'll come with some reagents and a protocol. And that protocol is really nice because without too much personalization, it seems to work almost every time, regardless of what you're doing differently or what cell type you're using that's different, and at least in my experience. So yeah, it'll give you mixes and basically all these holes in this chip where you're just adding different reagents into. And the only one in particular that I'm going to point out is this one here. So it says load cells, and it points to one particular well. So in the protocol, it'll tell you a particular concentration of cells to load, or a particular number of cells in a particular volume. And I'll start with volume because this is something you might play around with, although in my experience, I've kind of found a volume that I like and stuck with it, and I'll tell you what that is. But in the protocol, it'll tell you load between 5 and 20 microliters. Where the 5 comes from is the fact that even if you were to load a mil, which this thing can't fit a mil, but even if you were to load a mil, only 5 microliters is going to actually enter the chip. So, you know, the 995 microliters extra isn't actually doing anything for you. The 20 microliters just comes from the fact that this well can literally only fit about 20 microliters. So in my experience, I just concentrate whatever number of cells, depending on what the final concentration of cells that I want to be, I always just concentrate that in 6 microliters and load that whole 6 microliters onto here. 5 is going to go onto the chip, and 1 is extra so that no air enters the chip. And that seems to work well for me. Now, as far as what amount of cells, it'll tell you a particular concentration in that protocol. And I've actually found that that number it tells you, which I think is on the order of 200 cells per microliter, is actually, you can go higher and still capture single cells just fine. In fact, if you go higher, I've found that the higher you go, the more capture sites you get successfully loaded. So I wouldn't treat the number, if you do this experiment soon, I wouldn't treat the number in the protocol, the cell concentration number, as an optimal, but rather as a minimum, is what I mean to say. That being said, the more cells you load, the more you're risking multiple cells being captured in the same capture site, or a clog happening, although that is usually pretty rare, as long as the cells have been washed and you're not loading a bunch of ECM into the chip. But even like doubling the cell concentration that I load from what it says in the protocol, as long as I have EDTA in the suspension that the cells are in, remember these are epithelial cells, I seem to not have a big problem with getting multiple cells captured per well. But then again, the other variable there is that maybe that's only because the cells are perfectly sized for the chip that I'm using. So the number of cells and the capture site are kind of the two things to think about when you're trying to optimize how many single cells you get captured. Meaning, well, you're trying to minimize the number of capture sites that aren't captured, and also the number of capture sites that are multiples rather than singles, as long as you want to be able to call this single cell RNA-seq. In this microfluidic chamber, there are 96 capture sites, so everybody imagines on a perfect day they'll get all 96 of them loaded. In my experience, that's never happened to me. Even with cell lines, I think Angela has the record of 92. With primary cells, I've never gotten above 70. And if I get between 50 and 70, I'm feeling like it was a good day, and I'm moving on to the next step in the protocol. Now, if you load a first time, and basically when I say load, I mean...
Can I ask a question?
Please.
So, you said multiple cells in one capture site.
Yeah.
How can this happen? I thought, if the cell goes in, this is closed, so...
Right, so there's no...
Is it just because you don't have a single cell suspension and they still stick together, or
yeah, so like the EDTA thing I mentioned, that could be a reason. That's probably the more likely reason than if a cell's already captured, another one will stick there. Unless your capture site is just way too big for your cells, because there's no... When I say capture site, there's no like door. It's just, there's a little like divot in the plumbing, like a dimple almost. And if a cell lands there, it's the way that the chip is working is that capture site is literally not big enough to fit a second cell once a first one lands there. But there's no like door that shuts or platform that raises or anything like that that absolutely guarantees that a second cell wouldn't land there. Does that answer your question?
Kind of, yeah.
I think the cells, the issue of cells junctioning, especially epithelial cells junctioning together in suspension before they even enter the chip, is probably a bigger problem than cells junctioning once they're on the chip. So, yeah, if you're thinking about epithelial cells, use EDTA or use whatever trick you know of to keep cells from binding to each other too strongly. Okay, so once you've loaded cells into this, again, this here inlet, it'll take half an hour. You'll put the chip into this machine. I'll show you if you hit open. You put it in here, and it takes half an hour for the machine to pull the cells through the whole chip. So you just load it like this with the barcode facing you. And then if I were actually loading it, I would press load. But since I'm not actually loading, I'm just going to hit close. And you would there's a very obvious program that comes as a default program on these machines that says cell load. You have the option to also do a live dead viability stain. So if you add that dye into another inlet in the plate and you tell the machine that you want to load cells and do this viability stain, then it'll take an hour rather than half an hour. And that's another built-in functionality that this machine comes programmed with. So yeah, after that half hour, you'll take the chip out and you'll go over to, well, this is the microscope we use, but you'll go to some microscope to image that'll hopefully has an automated stage so that you can image all 96 capture sites in a reasonable amount of time. Since remember, this is for RNA-seq, and unless your microscope is in a cold room, then they won't be at 4 degrees for that period of time. And Brian's going to talk about the microscope setup that we have, so I won't go in depth there. But let's say you found, on the one hand, or the one option is you have found that you've loaded a sufficient number of cells to make you feel good about the experiment. Then you would come back and in 3, not the cell inlet hole, but in 3 other holes, you'll load 3 reagent mixes. One is for cell lysis, the next is for reverse transcription, and the last is for PCR. By default, it'll take the cDNA through 17 cycles of PCR to get you some amount of amplification so you can have enough material to make libraries. Those mixes do not come in the Fluidigm kit. We buy those separately from a company called CloneTech. And so once the cells are loaded and you've put in these 3 mixes, it's simply a matter of putting the chip back in the machine, telling the machine what program you want it to run, which is something called our RT-PCR, I think, is the name of the program. And that lysis, RT, and PCR step takes at the minimum 7 hours. So most of the time that's the last thing we do and run it overnight and then come back and get it the next morning. Now, if you didn't load it, if you found from the microscope images that you didn't load as many cells as you want, if you have more cells left over in whatever tube you took from in the first place, then you have the option to load cells again. That'll be another half-hour process. And there's a good possibility that you'll hopefully load additional capture sites that weren't captured in the first run, but also not double up capture sites that you already loaded in the first run. I found that it does actually work out pretty well that you load new sites without overloading too many old sites. Do you have a question?
You mean the second run load still use the same chip?
It's the same chip, but new cells. It cannot recycle the same cells that you ran through the first time. You have to put in new cells, but the same chip.
The same chip, and then it can just work on sort the new cell to the empty holes, right?
Well, it's not that it knows which capture sites are loaded versus unloaded, but you have the same physical process working in your favor as you did the first time,
okay,
where the capture sites are only big enough to fit 1 cell.
Okay.
Does that make sense?
Okay.
As far as one other critical thing is, so I forgot to say this earlier. If you want to get into that 50 to 70 cell, what I think is like as good as you're going to do for primary cells on these chips until some aspect of the technology changes, I found for my cell type with the chip size that I'm using, just to give you kind of like a ballpark, if I can't get, my cells are rare, so it's always like a possibility that I won't get this number of cells, but if I can't get 2,000 cells, 2,500 would be better, then I don't even load the chip, to be honest, just to give you like a ballpark number. So it's not that you load 96 cells and then some fraction of that 96 get loaded. You load a couple thousand, if not more cells, and then some number less than 96 of those will get loaded. Yeah, are there any other questions? I think that's...
You mentioned a problem of air bubbles. So
yeah.
I mean, respanning 2006 microliter, sometimes it might happen if you have some...
Yeah, so you might resuspend it in 6.5 microliters and then only take 6. When you're loading these things, yeah, it can be a little stressful trying to load them perfectly with no bubbles. If there's a bubble at the top of a liquid, that is usually okay because they're usually telling you the volumes that the protocol is telling you to load are usually a little bit more than the chip actually needs. So bubbles at the top are okay. Bubbles at the bottom, not okay, because those would be going into the chip first. Okay. Other questions? I don't think I have any other tricks of the trade. Okay. Thanks for listening.
So this is our fragment analyzer from Advanced Analytical. It's a important aspect to our operation here. It's how we can assess the cDNA quality coming off of the C1. We mostly just use it to look at the cDNA. It's basically a 96 sample bioanalyzer. which most people are familiar with. It works a little different. It uses gel electrophoresis. It's the same patent as the Sanger technology. Basically, it fills with gel in the capillaries and then electrophoresis through and generates here, images it right in here in this little laser box and generates these electropherograms and gel-like image from our 96 cDNAs. It only does 95 of them. Usually from the microscope images, you can pick out a negative and remove that and put the latter in for it to reference to. It has a upper and lower marker. Those are internal references for the assay. This one's a single base pair and this one's 6,000 base pair. We usually look for the cDNA in the upper range between 500 and 5,000. You can't really see it in this one because whoever's run this put their spike-ins in at a low concentration, but if you look at the gel-like image, there's these two bands that appear in basically all of them. Those are ERCC RNA spike-ins. They show you that the machine worked, that it pumped in the reagents, and then it pumped them out, and that this chemistry worked. Because if you just had a blank run, you wouldn't know if it's this machine or what had happened. So in addition, you get quantitative information. So the actual values coming off of this instrument are used going into the next era reaction afterwards. So generally, users will decide based on the images from the microscope and the images here. Sometimes they contradict, but usually they agree. And different people make a decision based on different criteria. But usually people want to have what looks like a single cell over there and what looks like cDNA over here. And the cDNA usually is over a certain threshold value that's enough to make a library out of. So this one probably has a couple dozen that could be made into libraries. You can see them real quickly. They're the ones that have a very bright band up here in the top part, which corresponds to the smear here in the top. In addition, we get the average size and the distribution of the sizing, which can be fed directly into an algorithm that could calculate all of this for you, which ones you want, and then also how to dilute them going into the next era reaction. We generally, after making the Nextera libraries coming off of here, pool them blindly. So we were originally, we would run the 96 Nextera libraries. We would make libraries off of these negative results, ones that look like this. We don't do that anymore. It's all ERCC, there's no point. We would sequence them too. But we would also then run the entire 96 samples from the libraries on here, but we don't do that anymore. We just pool them blindly. The reaction is very uniform, so any information you gain from here, you'd be mixing like 1.05 microliters of this one and 1.07 of that one, and you're basically just mixing 1 microliter of each one. And while this is cheap per sample, it's about $200 a plate, so no need to do extra runs for no reason.
So these are just examples of the flow cells that are used for next generation sequencing, and so this is the first one that we've had, the 454 system, and so this one is sequences on beads on these PGP plates. So the way that this works is that you have a bead and a single DNA molecule trapped in an oil emulsion, where it does emulsion PCR, where a single molecule is amplified up to several million copies to go on the sequencer. The 454 system and the ion torrent system are both based on basic properties of DNA replication. That is, when you incorporate a nucleotide into a growing chain, you release a molecule of pyrophosphate, which in the 454 system is coupled to a luciferase enzyme assay, which releases light, which is how you measure base incorporation. In the ion torrent system, the sequencing is done using these small semiconductor chips, which measures a proton being released. So it's really a series of small pH meters in there. So when you incorporate a base, you release a proton. The problem with these systems is that if you have like 5 A's in a row, it often only says there are 3 A's because the system can't handle these homopolymers very accurately. So there are other kinds of sequence-specific problems that it has. But these systems use normal nucleotides, they make normal DNA, and so in theory there's no limit to the read length in this system because they're not using any fluorescence or radiation or other kinds of modified nucleotides. So we also now have 3 single molecule sequencing systems. We have a helicose system, which was developed by Steve when he was at Caltech. And in this system, the flow cell has oligo-DT immobilized on the flow cell surface, and the DNA is tailed with terminal transferase and bound to here. And this also incorporates 1 base at a time and images the incorporation of a single floor in a single spot on the flow cell at a time. So the helicose system, they could never get the read length really beyond like 30 bases, and the way that their floors were designed, the DNA has a kind of a funny kink into it, and so their long sequencing time plus the short read length kind of let Illumina sort of eliminate them from the market, so to speak. So the PacBio system is still a single molecule system that's still flourishing. What they do is they take movies of fluorescent nucleotides as the enzyme is incorporating them. So this is a smart cell. This is what the single molecule sequencing takes place in small aluminum wells that are now about 150,000 of them get imaged in a single chip at a time. And so the way this works is when the enzyme takes a DNA into a fluorescent nucleotide into its active site, it pauses for a short period of time and sort of says, is this an A or a T? What am I going to put in here? And so when it gets to the right nucleotide, it holds it in the site for a longer amount of time. And so what they end up with is a movie, which is a trace of the time that the nucleotide spends in the active site. And that's what gives them their sequencing. They're not really measuring incorporation per se, they're actually just imaging nucleotide binding. So this machine weighs like 2,000 pounds. It occupies a special room with special air conditioning, and the smart cells, as they call them, come in these trays which get loaded into the machine. So the machine is actually fairly easy to run. The library prep for almost all the systems are based on platform-specific adapter sequences. So you have a double-stranded piece of DNA, whether it's a ChIP-seq molecule or a amplicon or a genomic fragment or an RNA-seq molecule, cDNA, which you ligate on, you in-repair ligate on platform-specific adapters onto it. And so this is the newest single molecule sequencer, the Oxford Nanoform Minion, or MinION actually as they pronounce it. And so this system plugs into a computer, and this is an example of one of the flow cells. So what it does is it has biological pores that send the DNA through the pore, and because of changes in the electrical current going through the pore, you get DNA sequence information off of it. The helicose system, at the time it was released, people didn't like it because it had a 5% error rate, which are primarily indels. The PacBio sequencing system has a 13% error rate, and they're primarily indels. But the Minion system right now has like a 30 to 25% error rate, which is everything from base substitution to indels. And so they just released their product a few months ago, and so they're still in their very early stages, but you can see the difference between sequencing on this machine and sequencing here. The MinION has a lot of promise, especially for sequencing like environmental samples and things like that. So PacBio has been very good at helping genomic assemblies and also doing full-length cDNAs, which is something that Illumina's short reads are really not good at. But primarily we use the Illumina system, and this display is kind of the evolution of the Illumina sequencing system from the early GA to the HiSeq to the MiSeq. As far as, so what Illumina's done here is when they issue a new platform, they often have an upgrade with a new flow cell, but they have the same footprint, but the flow cell has significantly more area to do sequencing, so their output is improved. And so the sequencer that we use primarily for the C1 RNA-seq is the new NextSeq system, and it has two outputs, a mid-run and a high output system, where the flow cells are, the lanes of the flow cells are just slightly smaller on the mid-run than they are on the high output. And so these two systems allow you to grow really big clusters on the flow cells, of which allows Illumina to use LEDs and cheaper cameras. So the cost of the HiSeq systems are like $600,000, $700,000, whereas this one is only like $250K. And these allow you to run one pool of many libraries at once, and it's designed so that it does two of the lanes do chemistry and two of the lanes do imaging at the same time, so that way the output is much faster.
How many samples does one lane hold?
Well, we use 384 indexes now, so you can multiplex, presumably 4 C1 chips worth of libraries on a single run if you want. And so behind you are like the reagent cassettes. These are the MiSeq and the MiSeq and the NextSeq. So you can see they're really very similar. This one is just expanded in size, where the reagent compartments are in the bottom, and this is a much smaller version of this. And this particular kit will give you between like 10 and 20 million reads, but this one will give you like 400 million reads. So that the new NextSeq actually makes the MiSeq look slow in proportion, and so we're very happy with our new system. So that's basically the whole sequencing tour.